Guys, welcome back to Unacademy NEET English channel. My name is Tina. Welcome back, guys. Welcome back. No, just a second. Just a second. All right. Voice will be absolutely normal. Right. Yes, please. So guys, in this session, as you can see, we are going to start with the Re-NEET preparation, because unfortunately, because of the failure of the system or whatever, now let's not talk about those things, but we are going to uh, prepare again for this Re-NEET exam. We are going to bounce back. See, I feel, there are certain things we cannot control in our life. So life may not be fair all the time. Let's learn it right now at this stage of life. Life may not be very fair all the time. You cannot control certain things, but you can always control your perspective. You can always control how you behave, right? So in this situation, guys, your perspective is going to change the game. I know there must be few students who thought that okay, now finally they have achieved what they have always prepared for and they wanted to, and they were scoring around 700 marks. They must be feeling frustrated, and there are other students whose exam did not go well. So for both the students, guys, we have this opportunity. Let's take it as an opportunity to do even better. Even if you were scoring 700, now this is the opportunity to do even better. So you have already seen the type of the exam, and now we are going to work on those shortcomings. We are going to revise the things. We are not at zero. We are not going to start from the scratch. We are already at a good level, and we are going to build a greater foundation onto that. So on that positive note, guys, we are going to start with our zoology part 1 of class 11. So I'll be completing the syllabus. I'll be completing the zoology of class 11 in 2 parts. Let me tell you. So in today's session, guys, I am going to discuss 3 very important chapters. Animal kingdom, we are going to discuss structural organization, biomolecules, frog and cockroach. These are the things we are going to discuss today. Voice is fine. Voice is absolutely fine. I don't know some of why are you saying that. Please uh, reconnect your headphones. Voice is perfectly fine, guys. It's absolutely fine from my side. Human physiology, Lakshita, will be taken in the next session. So let's start with the session, guys. Let's start with the session. And before we begin, let me tell you very clearly. So guys, this is a great opportunity now to work on a foundation, as I said. And for that, as we have always told you, Unacademy is by your side. So guys, Unacademy is bringing to you 700 plus hours of lectures, 30 plus tests, 15,000 plus practice questions, AIMT, that's all India mock test series, which is very important. Some of you felt that the paper was difficult because many of you have not taken the mock test, so don't make the same mistake again. Maybe this is a blessing in disguise for all those students who did not take the mock tests and found the exam difficult. So make sure that you take all these mock tests and all the YouTube Re-NEET series. You can join it for just ₹1. You have to click on the link given in the description box. And now further, without a further ado, guys, I am going to start with the first chapter, that is Animal Kingdom. Please get your NCERTs and copies ready. And give me a thumbs up that everything is ready and we can start. We can bounce back. Yes, guys. Give me a thumbs up that all is set and we can bounce back. Send me all the thumbs up and we can start with the, and this is going to be a detailed session, guys. This is going to be a very detailed session. Is that clear? This is going to be a very detailed session. Only today or question solving. So guys, question solving I'll be taking in the special classes. Don't forget to join me in the special classes also. So today's session will be from 7 to 12. Also guys, tomorrow and day after tomorrow, I know there were other classes scheduled, but unfortunately KK sir's uncle has expired, so he will not be able to take classes tomorrow. So today, tomorrow and day after tomorrow, I'll be taking the classes. KK sir will be resuming after Sunday. Is that clear? Okay. So on that note, guys, so our condolences to KK sir, and let's start with the session. So be ready with biology books today, tomorrow and day after. Timings will be from 7 p.m. 7 p.m. to 11 or 12 p.m. whatever it takes, we are going to complete the said topics. So today, as I said, animal kingdom, frog, cockroach, structural organization, that is animal tissues, and we are going to complete biomolecules. And in tomorrow's session, we are going to discuss human physiology. Is that clear? We are going to discuss human physiology tomorrow. So all set, perfect, perfect. Starting with, and this is going to be a detailed session, guys. I'm going to read NCERT. I'm going to discuss few questions. I'm going to discuss what type of questions NCERT is asked today. See, on that note, I want to tell you one more thing. Do you remember the saga of NEET 2024? What happened? NEET exam was conducted again. But did you analyze the paper? The second paper that they conducted was very similar to the previous paper. I am not sure that the next Re-NEET that NTA is going to conduct is going to be same or not, but at least we can't ignore what NTA has been doing, right? Doesn't use a lot of brains, to be honest, I feel. So, you know, it may be possible that they repeat the questions from this paper as well. So maybe, as they have done it in NEET 2024. So don't ignore this paper. Now, starting with the animal kingdom, guys, let's start. So animal kingdom, we know that Aristotle's classification was the earliest type of classification, right? I have taken this from biological classification. Wait for it. I have taken it from biological classification. Now see, Aristotle gave us two kingdom classification, right? Aristotle gave us two kingdom classification, but the basis was, and it was a scientific basis for classification, but what was the basis? For plants, guys, he took simple morphological features. And what was the basis of his classification of animals? The animals with RBCs and ones without RBCs. This is how Aristotle divided. And later on, it was Linnaeus who brought two kingdom classification. On the basis of what? If they have cell wall, they are they are plants. If they don't have cell wall, they are animals. But the the type of kingdom classification that we follow is given by Whittaker. R.H. Whittaker gave the 5 kingdom classification 1969, and see, we are going to follow this classification. And according to this classification, guys, kingdom Animalia has all the organisms which do not have cell wall. They are all heterotrophic. Now, before we go into the details of animal kingdom, you must know what are the basic features. They are all heterotrophic. They all have nuclear envelope, right, or in nuclear membrane. So they all are eukaryotic. They all are multicellular. Yes or no? They all have cellular, or they have tissue, or they have organ or organ system level of organization. These are the features of organisms that we are going to study today. Quickly let me know that these points are clear. So this has been taken from biological classification. And see, eukaryotic absent. So what is absent here? Cell wall is absent, membrane present, type of body organization, and mode of nutrition is heterotrophic. Now directly coming on to the animal kingdom. First of all, what was the basis of classification? These are the bases: the level of organization, body symmetry, germinal layers, coelom, segmentation, and notochord. These are the bases of classification. So on these many bases, we have divided kingdom Animalia into how many phyla? We have divided it into 11 phyla, and we know the names. I think there is no doubt about these. Let's see, I am going to touch every chord, no matter whether it is an easy topic or it's a difficult topic. I am going to revise it all. Okay, so stay patient. If you know these things, okay, this is an easy thing. Why ma'am is discussing? Because ma'am is going to discuss it for everyone, for all the students, everyone. Now see, so we are going to divide the organisms into these 11 categories. So basis of Classification I have already told you. It is the arrangement of cells, body symmetry, nature of coelom, patterns of digestive system, circulatory system, even reproductive system. All these things are going to be discussed here. Starting with the levels of organization, guys. Give me a thumbs up that you're all set. How many hours? Till, I don't know. Time is not the limit. Time is not the limit. We are going to complete these 4 things that we have already discussed. So levels of organizations, guys. There are 4 types of levels of organizations which are seen in kingdom Animalia. One is the cellular level of organization. Then we have tissue level, we have organ level and organ system. See what happens in cellular level. Body is still multicellular, but it is made up of loose aggregate of cells. How much? There is aggregation of cells, but it is loose aggregation of cells. It is just a republic of cells. So many cells have come together, but they don't form a tissue. Is that clear? So it is a loose aggregate of cells, and this is seen in case of Porifera only. So characteristic feature of Phylum Porifera. Then tissue level of organization. What happens? In between the multiple cells, interstitial fluid is present, intercellular fluid. Now tissue level is seen in Coelenterata and Ctenophora. Then organ level of organization is seen in Platyhelminthes. So organs started forming, not many, but few basic organs started forming. And then organ system level of organization started from Aschelminthes, that is roundworms, to Chordata. Give me a thumbs up that you read your you know all these things. So if you know these things, feel happy that okay, you're prepared well. Okay, now see, this is the NCERT line, loose cell aggregates. And what type of organization is this? Cellular. Now there is this very important line, some division of labor occurs amongst the cell. Not a lot of division of labor, but some division of labor is present in what? In cellular level of organization. Please watch these lines carefully. Now, the cells performing the same function, they form the tissues, and there comes the tissue level of organization. And which all organisms, which all phyla show tissue level? It is Coelenterata and Ctenophora. And then organ system is exhibited by members of organ level is exhibited by Platyhelminthes. Done. And then organ system is shown by Annelida, Arthropoda, Mollusca, Echino, Hemi and Chordata. They all show what? They all show organ system level of organization. Please let me know that these points are clear. And what type of organ systems do we see in them? Digestive system, reproductive system, nervous system, all will be seen in these phyla. Are these points clear? Moving on to now, see, if we talk about the circulatory system here, circulatory system are of 2 types. Circulatory system are of 2 types. One is open, see here, one is open and another is closed. Another is closed. See, what is open type of circulatory system? First of all, understand that in open circulatory system, capillaries are not formed. See, open circulatory system does have blood vessels, but capillaries are not present. Please understand that. So blood will flow in the open spaces. What happens? Blood in the open spaces, and those open spaces are known as sinuses. So blood fills the whole sinuses. Where are they found? Arthropoda, Echinodermata, Mollusca, Urochordata, they all have open circulatory system. And what about the closed and others also? Closed circulatory system will only be found in Annelida and Chordata, but not all chordates. So first time the closed circulatory system was found in Annelida. Now these can be the questions in the form of statements. Please understand. Then another type of system that we'll see is the digestive system. Now digestive system, if present, will also be of 2 types. See, first of all, Porifera doesn't have digestive system. Coelenterata, Ctenophora, they don't have digestive system. Platyhelminthes, if it is a parasite, it will not have a digestive system. But free living ones may have the digestive system, and that will be of incomplete type. Now, what is incomplete type of digestive system here? See, Platyhelminthes, but only which ones? Which are free living, right? So from Platyhelminthes, we'll have the incomplete type of digestive system, and from Aschelminthes to Chordata, we'll have complete. What does incomplete digestive system mean? There will be a single opening which will function as mouth as well as anus. But if we have two separate openings, for ingestion we have mouth, and for egestion, for exit of the undigested food, we have anus. These are the complete, this is the complete digestive system. Now you must know which phylum has which type of digestive system. Please let me know that all these points are clear. Do you have to make the notes again? No, not necessary. First of all, you will get these notes on my Telegram channel. Secondly, guys, it is all in NCERT. That is why I have brought NCERT clips along. You have to just mark them there. You already have your notes. Just use the same notes. Please use the same notes. Is that clear? Just keep revising things with me. Now, moving on. Second basis of classification is symmetry. What does symmetry mean? If the organism can be divided into equal halves or not. See, so we have two things. Either the organism can be divided or it cannot be. So if the organism cannot be divided into equal halves from across any plane, that is known as asymmetry. See, division of organism in two equal halves, and the two equal halves should be mirror images of each other. Now this is a key point. They should be mirror images. So if they can be divided, this is called symmetry. Now, which ones are asymmetric? That is sponges. Sponges are mostly, so if they say sponges are only asymmetric, no. There are few exceptions, although not given here, but there are few exceptions like leucosolinia. You don't have to learn that, but they can be radially symmetrical. So mostly word is a key word here. See, keep putting stars. See, this is where we make some mistakes, right? These have been our shortcomings. So those of you who felt excited with that paper and still missed on the keywords, these are the keywords. Sponges are mostly asymmetric. So any plane, be it any plane, it cannot be divided into equal halves. Is that clear? Second type, so that is porifera. Then we have another type, that is radial symmetry. Do you see a picture here? Now this represents the radial symmetry. What is radial symmetry here? Across any plane, going from top to bottom, you can divide the organism into two equal halves along any plane, and both the halves are mirror images of each other. Now this is the radial symmetry. Now very important, which all organisms are radially symmetrical? We have Ctenophora, we have Coelenterata, and the third one is Echinodermata. Now echinoderms, adults are radially symmetrical. Now this is the question here. That's why I've put a question mark. So they are going to ask you, echinoderms have radial symmetry only? No. Only the adults will have the radial symmetry, while the larva of echinoderm will have another type, that is bilateral symmetry. Are you getting that? So I have brought out the keywords. I'll be sharing these notes with you. You can revise from here as well. Now, talking about the bilateral symmetry. So, body can be divided into equal right and left only in one plane. And what is that plane? It is the longitudinal plane. That is why, guys, that is why bilateral symmetry is also known as longitudinal symmetry. So, starting from Platyhelminthes, moving on to Chordata, it is all bilateral symmetry except echinoderms, whose adults show radial symmetry, but their larva shows bilateral symmetry. So this means the larva is more advanced. More advanced. Please tell me that these points are clear to you all. Please tell me these points are clear to you all. Yes? Is that clear? I'll be sharing everything. I'll be sharing these notes on the Telegram channel. Don't worry. Yes, please. Now, moving on to the number of germ layers. So if there are 2 germ layers, the organisms will be known as diploblastic. If there are 3 germ layers, the organism will be known as triploblastic. Now see which ones are diplo and which one are triploblastic. So Porifera, Coelenterata, Ctenophora are the diploblastic. They just have 2 germ layers, which are ectoderm and endoderm. Right? Is that clear? While the triploblastic, as the name says, triplo, they have 3 germ layers. So they have ecto, they have endo, and the third one which develops is the mesoderm, starting from Platyhelminthes, moving on to Chordata. So Platyhelminthes are the first triploblastic organisms. Is that clear? I hope this one is clear. I hope this one is clear. Now see, in case of diploblastic organisms, see, outer one is ectoderm. This one is ectoderm. This green thing is the endoderm. And in between, do you see this pink colored structure? This is the mesoglea. Now this is not a cellular membrane. Please note down, why don't we consider it a cellular membrane? Because it is undifferentiated. See here, an undifferentiated layer, that is mesoglea, is present between this and this. It is non-cellular. It is non-cellular. It is not made up of cells. Since it is not made up of cells, we'll also call it non-living. Yes or no? It is non-living. Is that clear? So in diploblastic organism, ectoderm and endoderm is separated by a non-cellular and non-living layer known as mesoglea. Please tell me that these points are clear to you all. Please tell me all these points are clear to you all. Yes, in between is present the mesoglea. Are these points clear? So I hope you remember which phyla are diplo. The first three, Porifera, Coelenterata, Ctenophora, are what? They are diplo. Now, talking about the coelom. Coelom is the body cavity. Coelom is the body cavity. So not any cavity will be known as body cavity. It is a cavity between the body wall and the gut wall. See, this is very important. So, do you see here? Okay, let me show this again. Wait. Do you see here? This is the gut and rest. This is the body wall. And between the body wall and the gut is present this cavity, and this cavity will be known as coelom. This cavity will be known as coelom. Is that clear? Now, there are certain organisms which don't have the body cavity. See, there is gut, but there is no cavity around it. So these organisms will be known as acoelomates. Acoelomate, they don't have body cavity. Now, which all organisms are acoelomates? Porifera, Coelenterata, Ctenophora, and even Platyhelminthes. Now see, this is a case of Platyhelminthes. Why? Because they have drawn ectoderm, they have drawn mesoderm, and they have drawn endoderm. Right? There is no space. So it is triploblastic, but there is no body cavity. Hence, it is a Platyhelminthes. Now, even the diploblastics can be acoelomates. So these three, remember, Porifera, Coelenterata, Ctenophora, these are diploblastic and acoelomate. Note down very carefully, okay? And Platyhelminthes, these are triploblastic and acoelomate. So which diploblastic or which triploblastic is acoelomate, you have to note down very carefully. Instead of mesoderm, if they would have drawn mesoglea, then that would be the that would be the diagram of Porifera, Coelenterata, and Ctenophora. Please tell me these points are clear to you all. Now here, do you see? So outer one, this is the ectoderm. This one, this inner pink thing, this is the mesoderm. This is the endoderm. Now you see this pink layer around, this gray thing, this is the endoderm, right? And outside the endoderm and inner to the ectoderm, we have these two layers. Do you see this pink and this pink? So, if the body cavity is surrounded by mesoderm on both the sides, now please be careful. When do we call it a true coelom? This is the example of a true coelom. What is a true coelom? True coelom is the one when body cavity is surrounded by mesoderm on both the sides. Do you see? Mesoderm on the outer side, mesoderm on the inner side. This is inner mesoderm, this is outer mesoderm. So when it is surrounded by mesoderm on both the sides, this one is known as true coelom. True coelom, also known as eucoelom. Please tell me that these points are clear. Please tell me. Yes, please. Are these points clear to all? So see, the animals possessing the coelom, those which have coelom, are called coelomates. And when they say coelomates, they mean true coelomates. They are just using the term coelomate for the true coelomates. That means body will be divided by, this is true coelom. Body cavity will be divided by mesoderm on both the sides. We have outer mesoderm towards ectoderm and inner mesoderm towards the endoderm. Now, which organisms are true coelomates? Annelida, Mollusca, Arthropoda, Echinodermata, Hemichordata, and Chordata. Is that clear? Is that clear? So they are not considering pseudocoelom in the true coelom. They are not considering this. So only eucoelomates are the true coelomates. Is that clear? The third category we have is of pseudocoelomates. Now what is pseudocoelomates? See this diagram. Do you see these pouches? These are mesodermal pouches. These are mesodermal pouches which are scattered in the cavity. Is that clear? So since mesodermal pouches are scattered, hence the body cavity will be known as pseudocoelom. This is called pseudocoelom and it is a characteristic feature only of roundworms or Aschelminthes. Please tell me these points are clear. Please tell me these points are clear. Now pseudocoelom is a characteristic feature of, Aschelminthes. So can we say the body cavity is present? See, they have mentioned this cavity is the pseudocoelom. Is that clear? Can we say if the body cavity is not surrounded by mesoderm on the outer side or on the inner side, that is pseudocoelom? If mesoderm is present only towards one side, still it is a pseudocoelom. Please tell me that these points are clear to you all. Yes, please. Please tell me these points are clear to you all. Yes, please. Can we move on? Can we move on? Which is the coelom and pseudocoelomates? Which is the coelom and pseudocoelomates? Ninad, see this space. This whole space is the pseudocoelom. This empty space between, see this is the gut cavity. What does coelom means? It is the space between, it is the cavity between body cavity and gut wall. This is the gut wall. This one is the gut wall. This one is the body wall. And the space in between will be the coelom. But it is not lined by, hence it is a pseudocoelom. Is that clear? Now, moving on to, I have already told you there are two types of circulatory systems. Yes or no? Just discussed. So circulatory systems are of two types. One is open and second is closed. So which all organisms have closed circulatory system? I've just told you in which capillaries are present. So that is Annelida. Annelida is the first phylum to have closed circulatory system and the second one being Chordata. These are the only two which have closed circulatory system. Rest of the organisms will either not have circulatory system and if they have, it will be open type. And it is absent in. Also, it is absent in. See, it is absent in. Which all phyla don't have that? Porifera. We have Coelenterata. Then we have Ctenophora. Yes. Then what? Which all phyla will not have that? Ctenophora and also Platyhelminthes. Yes or no? So they don't have, and rest will have open type, and then Annelida and Chordata will have closed type of circulatory system. Is that clear to all? Is that clear? Please tell me, keep giving me thumbs up that you are understanding everything and you're able to revise all the things. Just a second. All the things. See, the volume is perfect. I made sure there is no tech glitch today. Okay, moving on to segmentation. Now, if the body is segmented or not. Now see, there are some organisms which show true segmentation. Now, true segmentation is known as metameric segmentation or metamerism. What do we understand by true segmentation here? See, if this is true segment, Now, there are certain organisms which are unsegmented. It's very important that you learn the examples very carefully. So the unsegmented organisms are, see, Porifera, Coelenterata, Ctenophora, many flatworms, Aschelminthes and Mollusca. They are unsegmented, completely unsegmented body. While there are organisms whose body is either truly segmented or it shows false segmentation. Now, false segmentation is also known as pseudo-metamerism. It is known as pseudo-metamerism. While true segmentation is known as metameric segmentation or simply metamerism. Metameric segmentation. Is that clear? Now, which phyla has true segmentation? Annelida and Arthropoda and Chordata, while pseudo-metamerism is seen in few flatworms, not all. Now, what do we understand by true and false segmentation? See, in true segmentation, guys, see what happens in true segmentation. In true segmentation, the number of body segments is fixed. Number of body segments remains constant from birth to death. So if we are born with head, thorax, abdomen, we'll die with these three. Keep aside the fact of accidental cases, but there are no segments that will form later on during the life. No. So the number of body segments is constant. Then internal and external segmentation coincides. Is that clear? So this is true segmentation. And the segments are known as metameres. The segments are known as metameres here. This is true segmentation. What are the body segments known as? Metameres. But in case of false segmentation, like in case of even tapeworm, number of body segments are not fixed. Tapeworm keeps losing the body segments and the new segments also keep developing. So the body segments are not fixed. Remember that. Each segment is independent of the other. Right? So external internal segmentation doesn't coincide, and the segments here, the segments in false segmentation, segments are known as proglottids. Known as proglottids. Are these points clear to all? Are these points clear to all? And basically you have to remember which phyla comes under which category. Please let me know that these points are super clear to you all. Yes. Moving on to the another, that is notochord. Okay. Now notochord is that one simple basis, that rod-like structure, which is going to determine whether the organism is a chordate or not. So out of the 11 phyla that animal kingdom is divided into, only one phyla, that is chordate, will have this structure. If the organism has this structure, it is a chordate. If it doesn't have, it is a non-chordate. See, notochord positive, then chordate. Negative means not present, it is a non-chordate. Which all are non-chordates? From Porifera to Hemichordata. What is a notochord? Now see that. Now see, this is the embryonic structure of a chordate. Now this is mouth, this is anus, this is the gut, alimentary canal, right? This is the ventral side and this is the dorsal side. Now do you see on the dorsal side, I have drawn this rod-like structure. This rod-like structure, guys, is known as notochord. It is rod-like, it is mesodermal in origin, which is very important to remember. It is dorsal in position and it is solid. It is not hollow from inside. It is dorsal. It may or may not persist. It may or may not persist later on. But if even once the organism has this structure, that organism will be regarded as a chordate. Please tell me these points are clear. It may or may not persist. Clear? So for example, you have heard of urochordates. The notochord is only in the larval tail. As the organism grows into adult, notochord is lost. Cephalochordates will have it throughout life, right? Talking about us vertebrates, in our case, it gets modified into the vertebral column. Then cartilaginous fishes, if you remember, they also have a persistent notochord. Yes or no? Please tell me. It only persists. See, it may or may not be persistent. Persistent means it may or may not remain throughout the life. But even if it is present for one moment in the life, the organism will be a chordate. Please tell me. And you have to remember that it is mesodermal in origin. You have to remember that. Is that clear? So in NCERT, it is written from Porifera to Echinoderms. Just add Hemichordata. Also, Hemichordata is also a non-chordata. Please tell me these points are clear to you all. Yes, please. Is that clear? Shall we move on? It is dorsal and it is the embryonic structure. It is only in the chordates. Clear to all? Please tell me. Now coming on to this table. Read this very clearly. See, Animalia. It consists of multicellular structures. We have already discussed that according to the Whitaker's kingdom. Now, all the multicellular organisms are known as metazoa. Unicellular animals are not included under the kingdom of Animalia according to Whitaker. Yes or no? Can you tell me, let's have a quick question also. Can you tell me which kingdom consists of unicellular animal-like structures? Yes, please. Which organisms have unicellular? Quickly let me know. Quickly let me know. Which organisms will have Protista, very good, very good. Ninad is the first one to answer. The unicellular organisms, animal-like structures, animal-like organisms, that is protozoans, will be found under the kingdom Protista. Very good. Now, see, kingdom Animalia, according to the level of organization, is divided into first cellular and rest, all of them have been put under tissue, organ and this. Right? Then, talking about the symmetry, cellular level and mostly asymmetrical. It is not written always, it is written mostly. These are the things that you have to take notice of during your exam also. But examination fear is a real thing, and that is when we forget these things, right? And those who take care, they are the winners. So, aajo. So, symmetry, mostly asymmetrical, body cavity absent. So, that is why it is acoelomate. So, which phylum that would be? That would be Porifera. Now, can you make questions yourself from this table? Think of it. If you are the examiner, a good examiner, not like NTA, you are a good examiner, can you make questions out of this table? Yes or no? Yes. If you have to identify which student is a good, intelligent student, can you make questions out of this? If you can think like that, you can always crack the exam. Think like an examiner, a good examiner. Now, see, tissue level. Now, out of all these levels of organization, there are organisms which have radial symmetry, while there are others which have bilateral symmetry. Now, this level, this radial symmetry, body cavity absent. What is the organism? It will be Coelentrates or Ctenophora. Yes. Now, tissue, organ system level of organization, but bilateral symmetry. Now, you know bilateral symmetry will start from Platyhelminthes. Now, from Platyhelminthes, those without body cavity, that is acoelomate, it is just Platyhelminthes, right? Then with false coelom, it is Aschelminthes, and with true coelom or coelomates, they are just calling coelomates, and they are these category. Please tell me that these points are clear. Now, why have they put a sign of asterisk star Echinodermata? Because they have written Echinodermata exhibits radial and bilateral symmetry depending on the stage. Now, quickly fill in this blank. Which stage will exhibit the radial symmetry and which stage will exhibit the bilateral symmetry? Quickly, yes. If you can answer the questions that I'm asking along, if you can answer the questions, you will obviously clear the exam. No matter what happened, just think, just bury it now. It is done. What has to happen, had to happen, has already happened. Keep answering these questions and we will clear the exam this time. Yes, 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 yes. So adult has the radial symmetry while the larva has the bilateral symmetry. Promise me that no matter what happened, whether you were scoring 400 or 300 or 600 or 700, promise me, promise yourself that this Re-NEET exam will be one of the best exams that you have given till now. Leave me a comment, do let me know, and promise me, write down, promise that this exam is going to be the bestest exam of your life so far. Life is long. You will give better exams, but this Re-NEET is going to be better. You have to write down in the comment section, right? Is that clear? Now, moving on to the next, we have this. See, we have already discussed this. Levels of organization, I have made this table. Germ layers, you know. So, diploblastic, these two are diploblastic. The first triploblastic is Platyhelminthes, and ever since it is triploblasts. Then symmetry, asymmetrical and mostly asymmetrical, right? Mostly asymmetrical, few will be radial, and then these are radial, both Cnidaria and Ctenophora, bilateral and so far. Is that clear? Now this table is just for your revision, just for your revision. Now see this question. What is an open type of circulatory system characterized by? Now what is the open type of circulatory system characterized by? Yes, please. It is characterized by pumped out of the heart and all the cells and tissues and directly washed into it. See, open circulatory system does not have capillaries. There are no capillaries here. Do you see? But it doesn't mean that open doesn't have blood vessels. Blood vessels are present. That means arteries or veins can be present. Arteries and veins can be present, but capillaries are not present. So blood From the heart, blood comes out into the arteries, and then blood is released in all the empty spaces. Now the empty spaces are known as sinuses. The whole body cavity or the sinuses are filled with the blood. So that is why, see, all the cells and tissues are directly washed into the blood. Do you understand this statement? Please see. Do you understand this statement? Yes or no? Do you understand this statement? Yes? Moving on to the next part then. Now we'll be discussing the details quickly. So I hope the pace is right, and now we are picking up the momentum. Now we'll be making it little more speedy. Okay. So starting with porifera, why do we call it porifera? Porifera means pore bearing. These are commonly known as sponges. These are pore bearing organisms. Generally marine, mostly asymmetric. Yes or no? Primitive multicellular. This is the first phylum of the multicellular organisms. So can we call them primitive? Yes. Now see, the level of organization we have read, it is cellular. Now what is the unique feature? Now see, all the pink boxes that I have put here, these are the unique features. We have already discussed the general features. Now the first unique feature is cellular level of organization. Unique feature is the one which you will not find in any other phylum. So cellular level of organization is only a characteristic feature of porifera, and we know what is cellular. There is just an aggregate of cells. It is a republic of cells, right? But it will not form the tissues, and it exhibits very basic level of division of labor. Clear? Then sponges have water transporter canal system. Now do you see? This is the water canal system. Let me show you here. Now this is the water canal system, guys. Do you see? There are pores present. These are the pores present. These pores are known as ostia. Clear? These are the pores or the openings, and on the basis of these pores it got its name porifera, right? Now from the pores, water is going to enter into these canals. These canals are known as incurrent canals. So through pores, water goes into these incurrent canals, right? And from the incurrent canals, it enters this area, and this whole area is known as spongocoel. Spongocoel is also known as the paragastric cavity. Very important. And then from spongocoel, water will go out. This opening is known as osculum, and then through osculum, it takes an exit route. Yes or no? So this is the route of water canal system. You should know the sequence. Guys, remember, from at least 2025, since 2025, NTA is loving these sequence-based questions. Be ready with that. Now, moving on to the next unique feature. So this is the water canal system. Do not confuse it with water vascular system, which is found in echinoderms. That is vascular system or ambulacral system. This is the canal system. So it has minute pores known as ostia, the central cavity known as spongocoel, through which water goes out, that is osculum. Now what are the functions? Of this pathway? See, it helps in food gathering. So water which is coming in is bringing the food along. Second, it will help in respiratory exchange. Third, when the water is going out, it is taking the excretory waste along. So it helps in the removal of waste. Now other things which are not written here, it will also bring the gametes. Is that clear? Now, third unique feature is conocytes. Conocytes are also known as collar cells. Now, how is a collar? See, collar is something that forms the boundary, right? Now, see the collar of this. Let's see the collar. So, collar cells are like flagellated cells, like this. They are present all around the spongocoel on the inner wall. Do you see that? So, all around we have these conocytes, the collar cells. They're forming the boundary. And do you see these are flagellated? Yes or no? So these are all collar cells. All are collar cells, right? They are flagellated. They line the spongocoel. That is very, very important. See, line the spongocoel and all the canals. Even within this incurrent canal, these collar cells must be present. But this is beyond the scope of this diagram. Is that clear? So what are these collar cells going to do? They maintain the water current, or they generate the, they generate the water current. Do you remember your childhood when we used to fill the bucket with water and try to rotate it? What happens? Water moves towards the periphery. So similarly, within the spongiocele, the water current will be generated, and that water current will push the food items or the gametes into this layer. Now guys, see, it has a very unique layer in between. It has a very unique layer. So here, between the outer and the inner wall, here is present a mesohyl layer. Do you see? It is written, it is a mesohyl layer. Now mesohyl layer will have so many different types of cells. So you need to push things into the mesohyl layer. Is that clear? Now who's going to do that? These collar cells are going to do that. Clear to all? Now digestion is intracellular. There are some cells present. There are cells in the mesohyl layer which are going to digest food, which digest the food, which are going to digest the food. So since digestion is going to occur within the cell, that is why we say it has intracellular digestion. That is not the case with us. It has intracellular digestion. Is that clear? Then talking about the skeleton is made up of two things. It is made up of spicules and it is made up of spongin fibers. Now see, what does skeleton look like? So there are spicules. Spicules are these structures. These are the spicules. These structures are called spicules. They can either be made up of calcium carbonate or they will be made up of silica. Please tell me that these points are clear. Please tell me these points are clear. Yes? Now, then there are some spongin fibers. Now these spongin fibers are made up of protein, and these two structures form the skeleton. Are these points clear here? Then sexes are not separate, so it means it is a hermaphrodite. We also call hermaphrodite as bisexual. We also call it as bisexual, right? So both sperms and ova will be formed within the same individual. Sponges reproduce asexually either by gemule formation. Now gemule is the internal buds, right? Or by fragmentation. There are two methods in which it is reproducing asexually, gemules and fragmentation. So you break a sponge, every part of sponge will form a new sponge. That is fragmentation. And then it also shows sexual reproduction by the formation of gametes. Now that is obvious. Then for fertilization is internal. Where does fertilization occur? It occurs here. This is the site of fertilization. So that means it is going to show internal fertilization. Where does internal fertilization occur? In the mesohyl layer. Yes or no? And then the development is indirect, means larva will be formed, and the larva is morphologically distinct from the adult. You have to remember that line. Please tell me, give me a thumbs up that these points are clear. Please tell me these points are clear. Yes, please. I have just joined. Should I continue from this or start from the very beginning? No, you can continue from here. You can continue from here. You can watch the basis of classification later on. Just go with the flow. Clear? All right. Moving on to the next, that is the example. See, first example is cycon, also known as scypha. Second example is spongilla. This is a freshwater sponge. And third example is that of Euspongia, and Euspongia is the bath sponge. You should know both the names, scientific as well as common name. They can easily come and match the following. Yes. Now, I am giving you a statement. What if I say all sponges are marine? Can we call sponges as exclusively marine? No, because spongilla is a freshwater sponge. So Porifera is not exclusively marine. It is mostly marine. It is mostly marine, but not exclusively marine. Yes or no? Please tell me these points are clear to you all. Please tell me these points are clear to you all. Yes, and you should be able to identify the picture also. See, this is the cycon, this is the Euspongia, and this one is the spongilla. You should be able to identify the pictures. Moving on now to the next, this is, just a second. Just a second. All right. Yes, guys. Moving on to the next phylum, which is Coelenterata. Coelenterata, also known as Cnidaria. Why do we call them Coelenterata? Because they have coelentron. Now, coelentron, the name is formed of two things. Coel means cavity, entron means intestine or gastro. So, gastrovascular cavity. Okay, and coelentron is also known as Cnidaria because of the special cells known as cnidocytes. Is that clear? Aquatic, mostly marine. Sessile means attached to the ground, and then the free swimming also. Radially symmetrical. The name Cnidaria is derived from the cnidocytes. Now, cnidocyte is a unique feature. Is that clear? Thank you. Now, cnidocyte is a unique feature, means it will not be found in any other phylum. See here, what are cnidocytes like? This is the structure given in your NCERT. What does it represent? See here. Let's see. What does it represent? This structure is the lid. This is a operculum or a lid or a cover. Now, this is one cell, while this one is the nucleus. These are some fibers which are going to hold it, and the most important part here, this part. This is that sac-like structure which contains a toxin. Now, this sac-like structure is known as a nematocyst. This toxin is going to paralyze the prey. So basically, see, cnidocytes or stinging capsules. Stinging capsule is also known as nematocyst. Where are they present? On tentacles and the body. What are they used for? Now, this is very important. You have to learn it 1, 2, 3, 4. What is the function of cnidocyte or the cnidocytes? One is anchorage, second is defense, third is capture of prey. So 3 functions. You can get questions like which of the following are not the functions of cnidocytes. So first of all, you should know what are the functions. So there are 3 functions: anchorage, defense, that means it is going to kill the organism, and capture the prey. So how does it kill the organism? So there is a toxin present in this stinging capsule or the nematocyst, and this is that thread tube which helps in discharging the which is going to discharge the poison, discharge the toxin into the body. So it represents tissue level, diploblastic, gastrovascular cavity, which is known as coelentron, and that is how it derives the name, and it has a single opening. Now see here. See, this is the cnidocyte when it is not used, and this was when it is used. Now I'm talking about the single opening, gastrovascular cavity. See here. So I'll draw it. Okay, I'll draw it here. So it has a vase-like body like this. Okay. Now there is single opening. This opening will function as both mouth as well as anus. So such alimentary canal will be known as, what? So this canal is known as gastrovascular cavity. Single opening mouth, and mouth is not directly present, so their mouth is present on the hypostome. Please tell me that these points are clear to everyone. Digestion is intracellular as well as extracellular. While in porifera, quickly let me know what type of digestion is present in the, what type of digestion is present, guys, in the porifera. Let's keep revising what we have learned. Type of digestion in the porifera, let me know in the comments, let me know in the chat quickly. Let me know in the chat what type. What type of digestion, guys? I'm not asking about the digestive system. I'm talking about the type of digestion in porifera. Intracellular. Yes, it is intracellular, right? And now, very unique feature of Coelenterata. Some of the coelenterata, not all, please take notice of this, only some coelentrates will form corals. They are known as corals. Why? Because basically, which coelentrates are known as corals? Which have calcium carbonate in their skeleton. So the skeleton is made up of calcium carbonate. Now what will happen? The organism when dies, the calcium carbonate skeleton remains behind. So only those coelentrates will be known as corals. Is that clear? Moving on. See, now, so this is also unique feature. Another unique feature is that coelenterata is not boring like us. It will show many avatars. It will have many body forms. So usually at least the two body forms are always present. See, exhibit two basic. It can be more than two. So the two basic body forms are, one is polyp and second is medusa. See the difference. See, medusa is this. This picture is from NCERT. This umbrella-like structure, which represents the jellyfish-like structure, right? This umbrella-like structure is the medusa form. It is free swimming. If you have ever seen the programs where they're showing different organisms, must have seen on Geographic or National Geographic or Discovery. So free swimming, jellyfishes are swimming, they're free swimming or free living. And now medusa form is responsible for providing defense. Is that clear? These are all tentacles, and tentacles have all those nidocytes. Is that clear? Now, medusa represents the sexual form. Why sexual form? Because it can reproduce sexually, it will form gametes and reproduce. While there is another form known as polyp. Polyp is more plant-like. Polyp means plant-like. Do you see? It is sessile. Just like plants are grounded, they are attached to one particular place. Polyps are attached to a particular space. Now, being plant-like, it is involved in providing nutrition. Now, polyp is the asexual form because it will reproduce asexually. Interestingly, both these forms can give rise to each other. See, medusa will reproduce sexually to form a polyp, and polyp will reproduce asexually to give rise to medusa. Now, this alternation of generation is known as metagenesis, and having multiple body forms, this is known as polymorphism. This is known as polymorphism. Yes or no? Do we exhibit polymorphism? No. No other organism does. It is only and only coelentrates. Is that clear to everyone? Please tell me. Now, here it is written, polyps produce medusa asexually and medusa forms the polyps sexually. For example, Obelia. Now, I was just going through one book, question banks. So it was written only Obelia does. No, it is not Obelia. So don't go just line to line. Understand. This will be done by all. Right? Now, what are the different examples? Physalia, very important example. And do you know Physalia has 5 different forms? So again, polymorphism. But at least 2 polyp and medusa will always be present. Portuguese man of war, Adamsia is a sea anemone, Penatula, as the name indicates, say pen, Gorgonia, easy to remember, sea fan, Meandrina, easy to remember, brain coral. Yes or no? Moving on to the next phylum is Ctenophora. They are commonly known as sea walnuts or comb jellies, or we call them sea gooseberries. Gooseberries, amla, they look like that. See the picture, it looks like a gooseberry. Now, this is the first phylum which is exclusively marine. Do you see this? Now, keep making a list now. Okay, keep making a list. You should be writing few points here. Write down on the top of your page, exclusively marine. First one is Ctenophora. As we go on, you will be writing down those names here on this list. Okay, moving on now. Yes. Exclusively marine, again, just like coelentrates, radially symmetrical, diploblastic, tissue level of organization. Another unique feature is, it has comb plates. What are comb plates? So these are the comb plates. So if I enlarge it, it has these teeth-like structures. These have, comb-like teeth. So basically these are not teeth, these are cilia. These are cilia. All the cilia are going to move in one direction and pushing the water backwards, and that's how it propels the body forward. So these ciliated plates help in locomotion. They help in locomotion. And how many comb plates are present? The number is fixed, 8 in number. Second unique feature exhibited by them is bioluminescence. That means it exhibits light. So while it is swimming at night, they look beautiful because they exhibit light. Property of emitting light. Is that clear? Which is well marked here. Sexes are not separate. So just like Porifera and Cnidaria and now this Ctenophora, they are all bisexual and only they reproduce only bisexual means. So they don't show asexual reproduction. Again, a unique feature. Fertilization is external. Fertilization is external in case of Coelenterata also. And guys, now after studying this chapter, chapter in this session, after doing all the chapters, let me know in the comment section if you want that one chart where the whole animal kingdom is summarized. Let me know with general features, with unique features, and with all the examples. Leave me a comment. Let me know. Let me know in the comment section. I'll share that with you in the Telegram channel. Is that clear? I'll share that in the Telegram channel, but after you have solved the questions with me, after you have solved these questions with me. Okay, so no spoon feeding. You will have to work hard. You will have to crack NEET with flying colors, no matter what. And I've told you, you have to promise this time, good or bad, whatever, it is gone. Let bygones be bygones. Now we have to focus here on Re-NEET, and we all have to get very good marks. Now, Pleurobrachia and Tinoplena, the commonly known as sea gooseberries because they look like gooseberries. Moving on to next. Now, Phylum Platyhelminthes. Why do we call them Platyhelminthes? Platy word for flat and helminths means worms. They are flatworms. Why flatworms? Because the body has been pressed and they are dorso-ventrally flattened. Not pressed actually, but it looks like they are pressed, right? So the dorso-ventrally flattened feature, and they're mostly endoparasites. Not all, mostly. Some are free living, and one of the examples of free living is Planaria. One of the examples here is Planaria. Planaria is free living, guys. Planaria or Deugesia is free living. It is not parasitic. Is that clear? So hence the word. Mostly endoparasites found in the animals including humans. They are bilaterally symmetrical. We have already discussed this. Triploblastic acoelomates and with organ level of organization. Now if they are endoparasites, then they must have some adaptations for that behavior. Number one adaptation. See, these are all the adaptations. These are the adaptations. They have hooks and suckers. Hooks and suckers will act as adhesive structures to attach to the body. They absorb nutrients from the body directly. Now this means there will be no digestive system. So the parasitic forms will not have digestive system. This is what I told you. Then specialized cells. Now these are the two parasitic adaptations. Other than that, there are specialized cells called flame cells. Now you will not find flame cells anywhere. So this is how flame cell looks like. See, it is a cell, and these are all cilia, which gives it a flame-like appearance, and hence the name. Otherwise, the flame cell will also be known as solenocyte. Flame cell also known as solenocyte. Is that clear? Now, fertilization. Now, this flame cell is important for osmoregulation as well as excretion. They are not separate. Sexes are not separate. Again, this is a bisexual. So far, we have not seen even a single phylum. Porifera, Coelenterata, Ctenophora, Platyhelminthes, all bisexual. Is that clear? Now see, fertilization is internal, which was the previous phylum that we have read, which shows internal fertilization. Let me know in the chat. Let me know in the chat which organism, which phylum shows internal fertilization. Quickly, let me know. Quick. Yes, please. Don't waste your time, guys. Let me know which Porifera. Yes, Ninad. Ninad is good. Ninad is the first one to answer my questions. That's how it should be. Very good. Excellent. I'm sure, Ninad, your paper is going to be Fadu. Great paper. Now, some members like Planaria. See, I told you Planaria is free living, and also it shows high regeneration capacity. No matter how many parts of a Planaria is cut into, every part will regenerate to form a new organism. Then the second example is Taenia or the tapeworm, and then Fistula or the liver fluke. Now both these are parasitic. This one is free living, told you, free living, and these two are parasitic. Is that clear? So fasciola causes liver rot. Why? And it is found in liver, of course, and tapeworm is found in our intestine and it causes teniasis. Is that clear? So this was about Platyhelminthes. Moving on to, now see, Platyhelminthes will either have unsegmented body, already discussed. Some of them will show pseudo metamerism, that is false segmentation. And do you see there are so many segments and the number of segments are not fixed. These segments, these are known as proglottids. These are known as proglottids. So they keep on shedding their proglottid and the new ones are added. Is that clear? And this is the fasciola, this is the liver fluke, these are their suckers through which they attach to the body of the host. Is that clear? Now moving on to the Aschelminthes, we also call them thread worms or we call them round worms. It is written, we call them round worms because the body is exactly round, as if somebody has used a compass to draw the diameter. Isodiametrically round, see, circular in cross section, hence the name. They may be free living also and they can be parasitic also. They can be terrestrial also, they can be found in water, that means they can be aquatic also. They are found in plants as well as they are found in animals. Is that clear? Now they have organ, first phylum to have organ system level of organization, bilateral symmetry, the only phylum to have pseudocoelom. Remember, scattered mesoderm pouches, yes or no? Scattered mesodermal pouches, do you remember? Yes, this is an English class. This is a complete English channel, but we keep English simple for everybody to understand. Yes? Okay. 5 minutes break? No. Arushi, no break. Not before this chapter. Now, alimentary canal. See, in Platyhelminthes, either it will be absent if it is a parasite and free living ones will have incomplete digestive canal. But Aschelminthes are the first one to have complete alimentary canal. So there will be a separate mouth and separate anus. Yes. Now in the alimentary canal, they will also have a muscular pharynx. Now this muscular pharynx is going to help it in taking the food. Is that clear? So excretory tube removes the waste from the body through the excretory pore. For excretion, they have a special excretory pore and the structures are known as actually Rennet cells. And other structures are called Rennet cells. Sexes are separate. So the first one to have separate sexes. Now males and females will start becoming different. And also not only they have separate sexes, they also show sexual dimorphism. So what is sexual dimorphism? That means you can distinguish between the male and the female from the outer morphology. Is that clear? See here. This one is male. How do we remember? The male is chota and mota. It is a shorter and broader version. While the female is slender and long and it has a straight end. Do you see? It has a straight posterior end. While the male will have a curved, this one is a curved posterior end. So this is the point of sexual dimorphism. 3 points: shorter and broader, slender and longer. Curved posterior end, straight posterior end. And now third point, these, do you see here? There are these structures. These are the penile setae. These are the penile setae which are only found in males and not found in females. Do you see these things? These are the points of sexual dimorphism. Please tell me these points are clear. So sexes separate, females longer than males. They are distinct. Fertilization is internal. Now keep making a list which all organisms have internal fertilization. Are you making that list along with me? Yes, I have asked you to make 2 lists. One is of exclusively marine. Second, I have asked you, now I am asking you to make a list of all the phylum which shows internal fertilization. Let me know in the comment section at the end that you did make these, and you can also share these with me in the on the Telegram channel. Let me know, are you doing these small efforts, because they are going to make a lot of difference. Porifera, Aschelminthes, that's it. So only Porifera and Aschelminthes have shown in internal fertilization till now. Please tell me that these points are clear. Very good. Pararia. Yes, absolutely. And what about Platyhelminthes? What type of fertilization was seen in Platyhelminthes? Yes, please. That was also internal. So those of you who have missed the Platyhelminthes, you need to recollect. You missed 1 point. So we have done 3 phylum which shows internal fertilization. Porifera, Platyhelminthes, Aschelminthes. Yes or no? Give me a thumbs up. How many of you remember that? How many of you remember that? Yes, please. Now see, the development can be direct as well as indirect. So larva may or may not be present and the young one resembles the adult. So that's the case with direct development. Now see the example. Number 1 is Ascaris. Ascaris is a roundworm, parasitic, and it causes the disease Ascariasis. We will discuss Ascariasis in human health and diseases. Second is Wuchereria, filarial worm, right? What does it cause? It is also parasitic. It causes elephantiasis. How? Now this Wuchereria enters our body through which mosquito? Please tell me. Which mosquito will bring Wuchereria? Through which female mosquito? Quickly let me know. How many of you remember? Let's see. And then third example is Ankylostoma or hookworm. All these 3 are parasitic. This also causes disease, Ankylostomiasis, not in your syllabus though. Are these points clear? Quickly let me know which mosquito is going to Culex. Perfect. Not Aedes aegypti. Puzzle, not Aegypti. It is Culex. It is Culex, female Culex mosquito. Moving on now, moving on to phylum Annelida. Annelida word comes from annulus. Word comes from annulus, which means rings. Which means rings. So aquatic, terrestrial, free living, sometimes parasitic. Also organ system level of organization, which has already been present since Aschelminthes. Bilateral symmetry, triplo, metamerically segmented, coelomate animals, body surface distinctly marked into segments or metamerism. Do you remember? The first phylum which has true segmentation. Yes? Now, I want you to note down certain things. There were certain things which appeared for the first time in Annelida. True segmentation, metamerism. True coelom for the first time. Longitudinal circular muscles which will help in locomotion for the first time here. And true or closed circulatory system for the first time. Please tell me that these points are clear. Please tell me these points are clear. And how many of you need that table in which everything is already present, already written? Yes, guys. Closed circulation. Yes, yes, yes. Me? How many of you want that? Yes. So everybody has to join me and remain with me here in the class. Attend all the chapters. Attend all the questions with me in the special classes and I'll share with you. Okay. Very soon on the Telegram channel. Perfect. Moving on now. Moving on. See, which helps in locomotion. Now, these Annelida can be terrestrial as well as aquatic. There are terrestrial as well as aquatic. Now, the aquatic annelids are, for example, see, you must remember the example of the terrestrial ones. See, this one is the aquatic. I'll write down here. This one is aquatic. This one is terrestrial. And this one is also terrestrial. Clear? Now, aquatic annelids like Nereus, they have lateral appendages called parapodia, which help in swimming. Very important. Do you see? This is an example of Nereus. These are the segments. Do you see? These are the small rings that I'm talking about. These are the rings we are talking about. And each segment is known as metamere. Clear? Now, this one is free living. This is also known as paddle worm because it has these parapodia. On every segment, there is a pair of parapodia present on each segment. These parapodia will act as paddles and will help in swimming. One pair on each segment. Please tell me these points are clear. Clear? Also, now, see, Nereus is an aquatic form and it is dioecious, means it is unisexual. While leech and earthworm, they are bisexual, hermaphrodites, while Nereus is unisexual. Please make sure that you're writing down all these things along with me in the when complete class. We are going to complete this 5 chapters. Yes. So guys, basically I take offline classes also till 5 o'clock. So I can only take online classes here on YT post 5. Okay, so I have taken time from 7 to 12. So we are going to take this for 5 hours and we are going to complete these chapters today. Now see, next, moving on. So I have to complete the syllabus in the offline, re-NEET for in offline also. See, so phylum Arthropoda. See, arthro means jointed, poda means feet. So jointed feet or jointed appendages. So unique features of Arthropoda which will always be present there. Not only feet, their appendages will be jointed. Now which all appendages am I talking about? I am talking about their legs, I am talking about antennae, and I am talking about their anal cerci. Now all these structures, they are jointed and their body has a chitinous exoskeleton, means it is made up of chitin. Is that clear? Now it is the largest phylum and 2/3 of all the named species are arthropods. So all the animal world, the whole animal world on one side and the arthropoda on other side, and they still form 2/3 of the whole animal kingdom. Think of it. Now organ system, bilateral, triploblastic, coelomates, chitinous exoskeleton, told you body consists of head, thorax, abdomen, but that is not always the case. It can be head, thorax, abdomen. Sometimes head and thorax will fuse to form cephalothorax also. Is that clear? Then they have jointed appendages, told you already. Now respiration through these structures, gills, book legs, book gills and book lungs and trachea. Now see, the respiration which occurs through trachea will be known as tracheal respiration, and you see that tracheal respiration in insects. Yes or no? Gills respiration is known as branchial respiration. And then book gills and gills, both branchial, and book lungs, this one will be known as pulmonary. Circulatory system will be of open type. Now the excretory structures, see there are different types of excretory structures as well. Malpighian tubules in insects. You should be majorly concerned about insects because you're going to study cockroach. Then coxal glands, then antennary glands, also known as green glands. Please tell me these points are clear to all, right? So it has huge diversity, this phylum has. So you'll also see diversity in different systems also. Okay. Now sensory organs like antenna, eyes, compound eyes will be both compound and simple. Statocyst, stato word is for equilibrium, right? So it will be a balancing organ. So these are the different sense organs. Antenna, remember, antenna is an olfactory receptor. You talk about that in cockroach also. Olfactory means meant for smell. Eyes are the photoreceptors. That is for light, right? And then statocyst for the balancing organ. Is that clear to all? Excretion I've discussed. Fertilized, see they are mostly dioecious. Mostly dioecious means some of them can be bisexual also. So mostly they are unisexual or bisexual. So cockroach, male, females are different, yes or no? So fertilization usually internal. Another list down. I hope you are making that list, everyone. I hope you are making that list. Yes, please. So another phylum which shows usually internal, but some can be external also. Is that clear? Mostly oviparous, development may be direct, indirect. Now economically important, honeybee, bombyx, lacifer, that is the lark insect. Then vectors means they are the carriers of the diseases. Anopheles, Culex and Aedes. Aedes will carry the dengue virus or the chikungunya virus. Chikungunya virus. And then also the flavivirus. And they all cause different diseases. While Culex is going to carry the filarial worm or Wuchereria and cause elephantiasis. We know that. And Anopheles carries plasmodium and hence causes malaria. Malaria is caused by plasmodium, but who brings that? It is Anopheles, and only the female versions, only the females will act as vectors. Gregarious means they always travel in big lot, that is locusta or locust. Living fossil means they have not undergone any evolution, no evolution ever since. So they are still the same. It is limulus or king crab. Please let me know that these points are clear. Now there are some separate examples given in the picture format. So this is a locust. This is the butterfly, of course. This is the scorpion. This is the scorpion, and this is the prawn, also known as paleomon. Please tell me these points are clear. Yes guys, please tell me. Arthropoda is done. Now moving on to the second largest phylum, which is Mollusca. Second largest phylum. And guys, yes, apart from these sessions, if you want to view or to watch all the 700 plus lectures, you want to take the AIMT, that is All India Mock Test Series, you have to subscribe to the Prime, which you can get, and you can watch all those lectures on Unacademy app just for ₹1 till your Re-NEET exam. Now this is a great opportunity. Make sure that you utilize this. I want to share something with you. I feel no matter where you are, one thing I've already said, we are not starting from scratch. Yes, we are not starting from scratch. We are already at a good level and we are going to build a better foundation over it. Make sure there is not even a single day till Re-NEET exam which is left or which is gone without you taking a mock test. Every day 2 to 5 p.m. should be reserved for the mock test. Not even a single day without mock test, guys. Take it as a challenge, and I promise you, if you do these things, nobody can stop you. Nobody, no system can stop you from clearing your exam. Is that clear? And that too with a great score, which is like 700, 650 plus, 700 plus. That is a great score if you follow these habits. Number 1, mock test every day. Secondly, you are revising those topics which are really weak. See, do one thing. Sit down with a calm mind without any grudges. What happened, what NTA did, what was fair, what was unfair, keeping that aside. I am sure we were not perfect, right? So sit with your paper and jot down all the weaknesses, all the shortcomings that you realized during your exam. And we all do. During our exam, we all feel, if I have studied that, why can't I remember this? Go through each and every question, look back the same day that you wrote that exam and look back and think where you made mistake, why you made mistake, what were you not able to recollect. List down all those things and revise. And do the same with every mock test. Not even a single day left without a mock test. Who will stop you from scoring 650 plus? But I know, no matter, it is so easy for me to say, not all of you are going to do that. And I challenge, no, not everybody is going to do that. So that is why, sometimes paper feels difficult, because we are not well prepared. Yes, every system is flawed and all those things, but let's talk about our flaws for now. This is an opportunity. How are you going to make a good use and a great use of this opportunity? What will create the difference? How will this NEET exam and how will this Re-NEET exam going to be different from the previous NEET exam? I'm not going to talk about the system failure. I'm going to talk about our flaws now, because this is an opportunity. So do these and let me know in the comment section that you are going to do that. I know it's easy said than done, and not many of you are going to do that. How many toppers are there? 1%? What makes that 1% topper? These micro habits, this discipline, and we don't have that, sadly. So let's see, and let me know in the comment section how many of you are going to do that. Talking about Mollusca, this is the second largest phylum, guys. Second largest phylum, terrestrial or aquatic, they can be both, and they can be found in both marine as well as fresh water. Organ system level of organization, bilateral symmetry, triploblastic, coelomates. This is what we have done. Body is unsegmented. And now they will have calcareous shell. Why? Because Mollusca is a soft-bodied organism. It is a soft-bodied organism. Now see here. The body is divided into distinct, although body is unsegmented, but there is a distinct head, then the visceral hump and the muscular foot. Is that clear? This is the head, and the head has these eyes, it has mouth, and the mouth has a special structure called radula, and we got a question in 2026 over radula. Yes, And then this is the visceral hump, which has all the internal organs. And this is a muscular foot, and it is very slow motion. It walks in very slow motion. Is that clear? Now, the body is soft, so there is this thin covering over it. The one in pink, this is mantle. Clear? This is mantle, very thin covering. Now, what does this thin covering do? This thin covering is going to secrete calcium carbonate, calcareous shell. What is made up of calcium carbonate is called calcareous. It is going, mantle is going to secrete. This is secreted by mantle. Is going to secrete the calcareous shell, right? Then there is this space between the mantle and the body, and this space is known as mantle cavity. Be very clear of where are these things located. Now, in this mantle cavity, do you see this white structure? These are the feather gills. These are the feather gills. Now see, a soft spongy layer of skin forms a mantle over the visceral hump, right? The space between the hump and the mantle. So between the hump and the mantle is the mantle cavity, which has feather-like gills, and these feather-like gills have two functions. One is respiration, second is excretion. So two functions. The anterior head has sensory tentacles, and then it has mouth, which contains a file-like structure. File means blade-like structure for rasping organ or feeding, and that structure is known as radula. Please let me know that these points are clear to you all. Please tell me. Is that clear? Is that clear? Okay. Now, moving on to the another part. These are usually dioecious. Dioecious means they are unisexual. So male, female, separate. They are oviparous. They lay eggs. With indirect development means larva will be formed. Do you see? These are two examples. This is Phyla, the apple snail. It looks like an apple, right? You might have seen these apple snails in the gardens if you go for a walk. And then this is an octopus, also known as a devilfish. These are the examples, very, very important. Phyla, apple snail. Pinctada is the pearl oyster. Sepia is the cuttlefish. Loligo, squid. Octopus, devilfish. Aplysia is the sea hare. Dentalium and Ketopleura. Third list I'm giving you. After doing the homework that I'm asking you to do, let me know in the comment section that you've done it. Let me know in the comment section that you've done it. Now see, the third list I'm going to ask you to make is write down all the names of the examples whose common names start with C. S-E-A-C. Sea hare. Sea pen. Right? What else? What else? Which all those examples whose common names have this prefix C, write down all the examples. And then in front of those examples, write down to which phylum do they belong to. It's going to be these small efforts which are going to make you a topper, guys. Now moving on to the next, which is Echinodermata. We call echinoderms. Echino means spiny. Derma means skin. So these are spiny skinned animals. Endoskeleton has calcareous ossicles. Ossicles are very small bone-like structures, not bones but bony structures. So they are spiny bodied, all are marine. Second example of the phylum which are exclusively marine. The first was Ctenophora. That list is also being extended, I guess. Second example is Echinodermata. Organ system. Now, the characteristic feature, adult and larva will show different type of symmetry. Otherwise, in all the other phylum, both adult and larva are going to show the same type of symmetry, but not in case of echinoderms. Adults will show radial symmetry, while the larva is going to show bilateral symmetry. They are triploblastic, true coelomates, digestive system is complete. So if this is a starfish, the mouth is on the lower side and the anus is on the upper side. So digestive system is complete. See, mouth on the lower ventral side and anus on the upper dorsal side. Distinct feature is a water ambulacral or water vascular system. In Porifera, we read water canal system. These have water vascular system. What are the functions? Now, this is a PYQ. This is already a PYQ. What are the functions? Number one, locomotion, capture and transport of food, and respiration. The question was, I think it was 2023 or 24. The question was, which of the following are not the functions of water vascular system? So if you know the functions, you will be able to answer. And I have discussed all these questions already before the NEET 2026 on 3rd May in all my classes. In the special classes also, I've discussed these PYQs. Yes, guys, please let me know. Please let me know that these points are clear. Excretion will come or not. Re-NEET confirmed? Yes, yes, yes. The whole world knows now. The whole world knows about it. Okay. Now see, this is the water vascular system. How is it? So the water enters through the special funnel-like structure. We call it medullary. Then we have the stone canal. You can skip that. But water enters into these canals. Do you see? These are the canals. These are the canals. So water flows through all these canals, goes into these canals, and through these canals into the radial canals, and through the radial canals into the tube feet. Now these tube feet are going to help in locomotion. Is that clear? Now excretory system is absent because the function of excretion is played by water vascular system. That is the 5th function of water vascular system. Sexes are separate, male females are different, reproduction is sexual. So fertilization usually external and development is indirect with free swimming larva. Now this is a second phylum where they have mentioned something about the larva. First it was about Porifera. So they have written that in Porifera the larva is distinct, morphologically distinct from the adult. Now it is writing that larva is a free swimming larva. Yes, what are the examples? Asterisk, starfish, Echinus, it is a sea urchin. See, those of you who find it difficult to remember and at least match the names, common names with the scientific name, see there is a hint. Echinus, it has U, it has urchin. Is that clear? Then Antedon is the sea lily. Cucumeria is easy to remember, sea cucumber, and Ophiura is also easy to remember. It is a brittle star. This is how brittle star looks like, and this is Asterias. Is that clear? Now moving on to the last phylum of non-chordates, that is Hemichordata. But now see, why do we call it Hemichordata and still keep it under the category of non-chordates? We still call it hemi, means half chordate, because it is half chordate because it shows one chordate like feature, which is pharyngeal gill slits. Pharyngeal gill slits is a chordate feature, but it is shared by hemichordates. Is that clear? But we still keep it under non-chordate. Why it is a non-chordate? Because it does not have a notochord. It does not have a notochord, right? But it does have a rod-like structure. This rod-like structure, which looks like notochord, but it is not a notochord. Why? Because it is endodermal. It is endodermal, and we call it stomochord. It is a collar-like region, and it is a in the collar region, it has this stomochord. Now, stomochord is found in the roof of the buccal cavity. It is found in the roof of buccal cavity. That is stomochord. While notochord we have read is mesodermal in origin. So, they're entirely different things. Earlier, actually, we did confuse it with chordates. We had kept it in chordate category, but later on we found the reality. This is stomochord, endodermal, not notochord-like structure. Is this point clear to all? Please tell me. Yes, guys. Moving on to the next, and we are going to start, okay, little bit more about hemichordates. Worm-like structure do we see? They are also exclusively marine with organ system level of organization, bilateral, triploblastic, coelomates, cylindrical body, and body is divided into 3 regions. One is proboscis, second is collar, and this whole structure is called trunk. This structure is called trunk. Is that clear? Circulatory system is open. Respiration through gills, excretion through proboscis gland. Now in this region, there is a proboscis gland which is meant for excretion. Fertilization is external, development is indirect. There are 2 examples, belanoglossus, secoglossus. Glossa means tongue. So the common name for hemichordata is tongue worm or a cornworm. Yes or no? Please tell me these points are clear. Yes? Now talking about the phylum chordata. Phylum chordata. So what are the unique features of chordata? Number 1 is notochord. We have already talked enough about notochord. Then there are gill slits in the pharyngeal region. So pharyngeal gill slits, and this feature is also shared by hemichordates. Now these gill slits will form gills later on. In our case, these gill slits would have been present here, but they got closed, and in our case lungs develop, right? We don't have gills. Otherwise these structures will give rise to the gills. Now then, first is notochord, second is gill slits. Then we have a nerve cord. Now nerve cord was also found in the non-chordates. Nerve cord is found in the non-chordates. The difference is, in our case, that is chordates, nerve cord is dorsal. It is single, it is hollow, and it is ectodermal. While in case of non-chordates, the nerve cord would be, now just compare, nerve cord would be double, it would be double, it will be ventral, it will be solid, with so many ganglia over it. Do you remember? Please tell me, can you compare? This is the nerve cord of non-chordates and this is the nerve cord of chordates now. So all these structures do we see? And the fourth structure is post-anal tail. This post-anal anal part, this is the anus, this is the mouth, and the region behind anus is known as post-anal part, also known as tail. Please tell me that these points are clear to you. Yes, please. Quick. Please let me. These points are clear. Give me a thumbs up. Guys, we have reached chordates. Quickly tell me. All these points are clear to you all. Now this is a table given in NCERT. See, notochord present, then chordate, absent, then it is a non-chordate. Nervous system or the nerve cord will be dorsal, hollow and single, while here will be ventral, solid and double, just discussed. Pharynx, this pharynx region has gill slits. These gill slits are absent except in hemichordata. Yes or no? Except in hemichordata. Now heart is ventral. This is the side where we have our heart and we feel the heartbeat. In non-chordates, if it is present, it will be dorsal. Is that clear? Then post-anal part, tail is present, it is absent. There are other differences also, like chordates will have RBCs and platelets. These will not have, and many more. Please tell me all these points are clear. Solid means it is not hollow from inside. So if we see the refill of the pen, that is hollow. That's where ink fills in, right? Solid means there is no space in between. Clear? Now, moving on to phylum chordata. See, phylum chordata is divided into 3 subphylum. Also remember the taxon. Remember the taxa. So phylum chordata is divided into 3 subphylum. One is Urochordata, second is Cephalochordata, and third is Vertebrata. Now let's decode the name. Urochordata. Uro means tail. So chordata. So notochord is only found. Uro means tail. So notochord only in the larval tail. Is that clear? Now second, Cephalochordata. Cephalo means head. So in Cephalochordata, notochord is present from head to tail, and not only in the larva, this is present throughout the life. I told you notochord can be persistent or it can be non-persistent. So here it is persistent throughout the life. Here it is only found in the larval stage. While in case of vertebrates, see, in vertebrates, notochord only in the embryonic stage. Later on it modifies to form the vertebral column, and that is why the name is vertebrates. Please tell me these points are clear. Please tell me these points are clear. Right? Now see, urochordates and cephalochordates are put under one category, that is protochordate. Proto means primitive. These two are the primitive ones, while vertebrates are called the true chordates or the modern chordates. Euchordates. Is that clear? Also guys, protochordates do not have cranium, that is the brain box. So we also call them acraniata. While vertebrates have brain box, they are called craniata. Is that clear? Then another difference. See, Urochordates will have open circulatory system, while now rest of them will all have closed circulatory system, starting from cephalochordates. Usually we say that chordates have what type of circulatory system? Closed. But the exception is they have open circulatory system. Are these points clear? Are these points clear? Now see the example. See the example. Urochordata, we have Acidia, Salpa and Dolulum. Acidia, Salpa, Dolulum. This is the picture of Acidia. Is that clear? Now, these two structures that you see, these are the siphons through which water enters and then exits. And it is sessile, means it is attached to the ground. And then cephalochordata. Cephalochordata example is Branchiostoma. Just one example given, also known as Amphioxus or the lancelet. Please tell me these points are clear to you all. Please tell me. Right? All right. Moving on to the next now. Now we are going to talk about vertebrata in detail. Vertebrate possess notochord during embryonic and later on replaced by the vertebral column. That is why vertebrate. Now this can be bony or cartilaginous. Now this is one famous line which you will never forget throughout your life, no matter whether you become MDS or MD doctor. But this line stays with you all throughout. See, now all vertebrates are chordates, but all chordates are not vertebrates. Can we say that? Yes. So all vertebrates are chordates, all chordates are not vertebrates. Now they have a ventral muscular heart on which side? On which side? It is on the ventral side, right? And then kidneys for excretion and osmoregulation, and paired appendages, they can be fins or limbs. Please tell me these lines are super clear. Another picture. Now see, this is a subphylum. This is the third subphylum. Now subphylum has been divided into two divisions. Now on what basis? On the basis of jaw. What is the basis? The basis of jaw. See, those which don't have jaws around their mouth, we call them aganetha. Ganetha means jaw, a means absence of jaws. So we call them jawless fishes. And then there is ganetha, ganethostoma, that means jaw around the mouth. Is that clear? So on the basis of jaws, there are two divisions. Now aganetha, jawless fishes, we have one class, that is Cyclostomata. They will be jawless. And they have cyclostoma, means mouth, cyclo means circular. They have circular mouth, they are jawless. So how will they take in the food? They will suck food. So they have suctorial food. Is that clear? Circular, suctorial and jawless mouth. Is that clear? And then ganethostoma, that means they have jaws. They can be divided into two superclasses. On what basis? On the basis of locomotion. On the basis of locomotion, or you can say locomotory structures. Locomotory structures. See, those which have fins are known as Pisces, and those which have four limbs are known as Tetrapoda. Is that clear? These are the true fishes. So ones with the cartilaginous endoskeleton are called Chondrichthyes. Those with the bony endoskeleton are known as Osteichthyes. And then we have four classes here: Amphibia, Reptilia, Aves and mammals. Let's discuss them one by one. Talking about Cyclostomata. So as I told you, they will have circular mouth, right? See, suctorial circular mouth without the jaws. This is what the name says. All living members are of the class Cyclostomata are ectoparasites. Very important line. They all are ectoparasites. They mean they will be attached on the outer side of the fishes to get the food, right? They have elongated body in how many pairs of gill slits? 6 to 15 pairs. Now make a list. There's the fourth list I'm going to ask you to make. This is of Cyclostoma. Then we'll talk about the Chondrichthyes. Okay, and then the Osteichthyes. Just write down somewhere at the top of the page and write down the number of gills present. This is what you are going to do. Make it right now. Here in Cyclostomata, it is 6 to 15 pairs. Noted? Now wait for Chondrichthyes to complete that list. So they have circular suctorial mouth without jaws, also devoid of scales and paired fins. That means devoid means they're lacking scales as well as paired fins. So they have fins but not the paired fins. Fins are not in pair. Cranium and vertebral column are cartilaginous. Okay, and circular system is closed type. They are marine. Another examples, exclusively marine. They migrate to spawning to fresh water. So basically what happens, adult is going to swim, this is the adult is marine, and it goes to the fresh water. It goes to the fresh water for spawning. That means for laying eggs. Is that clear? After spawning, after laying eggs, they are going to die. Now, the eggs that they have laid, these eggs will hatch. The larva will come out. The larva will undergo metamorphosis. And after metamorphosis, it is going to swim back. It will become adult, and becoming adult, it will come back to the seas. Yes or no? So this part occurs in the fresh water. This is in the fresh water, while this occurs in the marine water. Please tell me that these points are clear. Please tell me these points are clear, and make sure that you make this table, guys. 6 to 15 pairs of gills or gill slits. Example, this is petromyzin or the jawless vertebrate. Do you see this? Ventral, suctorial, circular, jawless mouth. Do you see these gill slits, 6 to 15 gill slits, and they are paired, present on both sides? Do you see the fins present, but they are unpaired? Do you see the whole body is smooth? It is scale-less, devoid of scales. Another example is of myxine. Now both these are PYQs. You have been asked to match the scientific name with the common name. Please let me know that you are done till here. Any doubt? Any doubt till here? Yes, guys. Any doubt? Quickly let me know. Yes, guys. All clear? Give me a thumbs up. Now we all need an energy break. Yes, guys. I need an energy break. I need lots of thumbs up. I need all of you to share this session with your friends. I want you to complete these series. Let's prepare together, guys. There is not even a single chance that we cannot score 360 out of 360 in biology. And of course, in physics and chemistry also, we can get wonderful score. Just follow these series. Everything is free. It's not necessary that, the good things are always have a price tag, and that too a big price tag. No. All clear? Let's have a water break. Not going anywhere, just a sip of water and coming back. Wasim sir mentioned that classes will be taken from the basics. Yes. So he mentioned it absolutely right. Don't you see that we are discussing everything in the basic, from the basic to the topper level? Yes. How to avoid burnout? Just stay hopeful in life. You're too young to be hopeless. Yes. Life is long. There are good things going to happen. And of course, don't assume that life is always going to be fair. That's not how life is. I can tell you, there will be certain things which are beyond your control, but that's how it is. And a part of growing up is to accept it. Sadly, badly, happily, whatever, but that's how it is. Okay. How to prepare? Burnout can also be, guys, prevented by staying in touch with your teachers. That is also another way out. Talk. Don't stay, introvert all the time. Talk. You should have someone to talk to. You should always have. Parents. If not parents, then maybe your seniors. If not that, maybe your brother, sister. If not that, then 100% your teachers. Talk to us. You have that facility to talk to us in chat, in comments. I always read comments. In special classes, you can talk. Always share how you feel. That is very important. See, that is why we keep sending those videos to you, because we want to talk. It's not the camera that we are talking to. We are talking to you. So you also talk to us. Those old days when we used to write letters. These days we write comments. So let me know how you feel. So whenever you feel burnt out, write. Send us those messages. We'll read, we'll revert back. Now, moving on to the next. That is chondrichthyes. Now we are going to talk about the gnethostoma. Now see, Chondrichthyes, they are marine. They are marine with streamlined body, cartilaginous endoskeleton, mouth located on the ventral side. See, whenever you think of Chondrichthyes, think of a shark. We all have seen a shark in TV, right? We all have seen a shark. See, the body, the mouth is on the ventral side. It has teeth which are backwardly directed teeth. Backwardly directed teeth. Yes, mouth is ventral. Do you see gill slits? How many gill slits are present here? Gill slits are separate. Yes, guys, how many? And you see the gill slits because they are not covered by the operculum. They are not covered by the operculum. Now, the scales present on the body are plecoid scales. Remember, and you always see one of the lobe is larger and the other lobe is smaller. So the tail is not homocircle, it is a heterocircle tail. One long one large and one shorter lobe. So notochord is persistent. Now this is another feature to be noticed. Notochord is persistent in two categories. One in the Cephalochordata and second in the Chondrichthyes. Yes or no? Yes or no? Clear? Then do you see gills are separate and they are without operculum or the gill cover. The skin is tough, containing minute plecoid scales. Teeth are, and these teeth are nothing but modified plecoid scales. Modified plecoid scales, right? This is written. Backwardly directed, told you. And they are very powerful. Jaws are very powerful. Think of the sharks. They are the predators. They are one of the most powerful predators. Once a prey comes in their contact, there is no escaping from their powerful jaws. They are predacious. Their absence. Not, have you ever seen a shark not swimming? No. Why? Because they don't have air bladders. They don't have air bladders, so they have to constantly swim to avoid sinking. Isn't that interesting? They have to constantly swim to avoid sinking. Please tell me these points are clear. Ma'am, where to attend the special classes, where to attend special classes, where to attend special classes. Jyoti, special classes, special classes, special classes. Okay, special classes have to be attended on Unacademy app, learners app. So just make your profile, and these special classes are for they are absolutely free. You have to just follow me on the Unacademy app also, and so that whenever I align a class, you get notified, and then we can practice the questions together. There were 8 questions which were repeated from my special classes. I'll be sharing those 8 questions with you. Okay, I'll be sharing those 8 questions which we have already discussed in special classes, and they came in NEET exam, and I have full proof for that. I've not made a video for that, otherwise exact same questions, and not those questions which were just somewhat similar. No, exact 8 questions. Now see, other things. Heart is 2-chambered in all the fishes, 1 auricle and 1 ventricle. And some of these fishes, see, and this is the circulation of the fishes. Fishes have single circulation. So the blood which gets pumped into the heart doesn't enter back. So it is a deoxygenated blood which enters the heart, and then it goes directly to the gills. Gills are going to add oxygen to it, so it becomes oxygenated. Now oxygenated blood will directly move into the body directly, right? So this is single circulation, and only deoxygenated blood enters the heart. So heart is venous type. This heart carries only deoxygenated blood. So is that clear? So it is a tube-like heart, it is a ventral heart because it is a chordate, and it is a venous heart because it contains only deoxygenated blood. Are these points clear? Now some examples. Electric organs have special structures seen in torpedo. So we call torpedo as electric ray. We call torpedo as electric ray. And what are these electric organs? These are modified muscles. These are modified muscles for their protection. Then trigon has a poison sting, and both these are PYQs. Then all these fishes are cold-blooded. That means their body temperature is not going to remain constant. They are poikilothermal and they lack the capacity to regulate their body temperature. Sexes are separate. Male, females are different. Now, how do you identify there is a sexual dimorphism? Now, males will have pelvic fins with claspers. So, there are some clasper, means claspers are to clasp, to hold. These claspers will be found only in males to hold the female during the time of copulation. They show internal fertilization. Are you listing down all the phylum which show internal fertilization? Yes, please. Are you listing down? Are you listing down? Please tell me. Yes, please. Are you listing down? Oh no, volume is absolutely normal. Why are you writing it again and again? It distracts me. I sometimes feel, okay, maybe students are not able to hear properly. I am shouting. Now, many of them are viviparous. Viviparous means they will give birth to the young one. Not all, just many of them. Is that clear? Now, moving on to the examples. Dogfish or scoliodon. So, this is the scoliodon or the dogfish. Okay, scoliodon. Clear? Do you see their heterocircle tail? Right? Now, do you see this is the mouth, ventral mouth. Pristis, this is the pristis. Pristis, it has a saw-like mouth. So that is why we call it sawfish. Carcharodon, that is great white shark. Trigon, the stingray. Torpedo, the electric ray. Just discussed. Please tell me these points are clear. You should be able to identify the pictures given in NCERT. Okay? And we will practice the such questions also, picture-based questions, all in my special classes. And as well as on this channel. But on the channel, my first priority is to complete the whole syllabus. But side by side, my question practice will be going on in the special classes. So don't forget that. It is all on the app. And if you want to watch the you want to practice more questions, then guys, just link is given in the description box, and you have to subscribe to the Prime membership just for ₹1. Okay, all right. Moving on to the Osteichthyes, that is the bony fishes. They can be marine as well as freshwater. They can be marine as well as freshwater. Bony endoskeleton, body is streamlined in both, mostly terminal mouth. Now think of this. This is a typical bony fish. So terminal mouth, do you see here? No, you don't see the cuts, gill slits. Why? Because it has a gill cover. Operculum is present, gill cover. Now, the type of scales present in the body, they are cycloid or ctenoid scales, and the tail has both the lobes absolutely equal. So we call it homocircle tail. Is that clear? Then operculum present, I've told you about the scales. Now you will see these fishes static in water. Okay. So why are they able to not swim and remain stable in water at a particular level? Because they have a swim bladder or the air bladder. See, air bladder regulates the buoyancy. Heart is again two-chambered, absolutely same. Sexes are separate. They are again cold-blooded. Now this is usually external. What did we say about these? It has internal fertilization. It has internal fertilization. While bony fishes have usually external fertilization. They are mostly oviparous and development is direct. Please tell me all these points are clear to you all. Please tell me. Yes, please. Yes, nothing. Tomorrow we will complete a human physiology, total human physiology chapters. Is that clear? All right, guys. So these are the examples. Now see which ones are the marine, Exocetus or the flying fish, Hippocampus or the seahorse. Now this seahorse is a male seahorse because it has a brood pouch. Now male seahorse does not lay eggs. It will carry the eggs which the female has laid in the brood pouch. Is that clear? So eggs will hatch, larva will come out from this. So here male shows the parental care. Now flying fish, why do we call flying fish a flying fish? Because it has a well-developed pectoral fin. Well-developed pectoral fin. So, you know, you see fishes coming out of water, they're gliding out of water like this. Now this particular fish, flying fish, because it is a large, very large and very pretty pectoral fin or the shoulder fin, so it is able to glide, have a bigger glide, so it is able to stay out of water for a longer time. So it looks as if it is flying, otherwise it doesn't fly. Is it clear? Well developed pectoral fin. Then freshwater ones are labio, catla, and clarius or magur, and those examples which can be put in aquarium, that is betta or flying fish or pterophyllum or the angelfish. Now please tell me these points are clear. Please tell me these points are clear, all of you. Yes, yes, yes. Moving on to amphibia. Now, with amphibia, we start with tetrapoda, that is those organisms which have 4 feet. Now, amphibia live in aquatic as well as terrestrial habitats. They have 2 pairs of limbs. 2 pairs means 4. Tetrapoda, body divisible into head and trunk. We will be discussing frog today and you will see all these features in frog. Amphibian skin is moist without scales, so does the frog have. Eyes have eyelids. In frogs also. And in frogs, the lower eyelid is movable. Our upper eyelid is movable. Frog's lower eyelid is going to be movable. Now, tympanum represents the ear. The pinna is a unique feature of mammals. But pinna is not present in all the mammals. Okay, please take notice of that. There are certain questions, which of the following features are present in the category associated without any exception. So pinna is not present in all the mammals, but only found in mammals. Please tell me these points are clear. Is this clear? Now, see, there was a question on this point. Alimentary canal, urinary canal and reproductive tracts open into a common chamber called cloaca, which opens to the exterior. The question was the female oviduct, female frog, oviducts and ureters open separately in cloaca. Okay, while separately we'll discuss in frog. That was a question related to frog, because in frog, male frog, urinary and reproductive tracts are going to converge. They'll be similar. Now, respiration is by gills, lungs and through the skin. Now, see here. It respires through skin. See, the tadpole or the larva, that is the tadpole, respires through gills. And what is the respiration through gills known as? Branchial respiration. So, tadpole is fish-like and shows this fish-like feature, respires through gills. While the adult, now see the adult, when it is underwater, it respires through skin, and the respiration through skin is known as cutaneous respiration. While on land, it uses lungs as well as mouth. So respiration through lungs is known as pulmonary respiration, and that through mouth is known as buccopharyngeal. This was a question? Yes or no? Buccopharyngeal. So this is how I've shown you the cloaca. Now, talking about the heart. Fishes have 2-chambered heart. Amphibians will have 3-chambered heart, 2 auricles and 1 ventricle. So this is how their heart is. So we'll talk more about the heart of frog also. So see this. So they have right auricle, left auricle and a common ventricle. So on the right, from the right side is going to enter the deoxygenated blood, left side will bring the oxygenated blood, and in the ventricle the 2 bloods will undergo mixing. Is that clear? So because the most of the body receives the mixed type of blood, so it is not able to regulate the body temperature, hence it is a cold-blooded animal. Is that clear? Then sexes are separate. Male, female frogs are going to be different. Why am I saying frog? Because we have to read frog in particular. And frog is a representative. You think about frog, you can write about amphibians. Is that clear? External fertilization, oviparous, development indirect. You have seen the tadpole is the larval stage. Please tell me all these points are clear. Yes, guys. These are the examples. Now, this lizard-like structure is not a lizard. It is a salamander, and salamander, guys, is an amphibian. And this is our rana, which is a frog, Rana tigrina. Clear? Other than that, we have bufo, which is a toad. Hyla is a tree frog. Ichthyopsis is the limbless amphibian. It is a limbless amphibian. And see, I've already told you about the heart, and it shows incomplete double circulation. Fishes have single circulation, they will have double circulation. But because the 2 bloods undergo mixing, that is why it is incomplete circulation. Please tell me these points are clear to you all. Yes, guys. Please tell me these points are clear to you all. Yes or no? Quick. Are these points clear? What is sinus? We'll come on to that. Arushi, we'll talk about sinus. Yes. we will talk about sinus in frog. Okay, if you're talking about that. All clear? Can you tell me all the table? Ma'am, can you please tell me all the table? Which table are you talking about, Sankalp? Which table? The table that I have to give you, which I have promised you. If you complete the whole session, if you attend all the special classes, you practice questions, that table or the one? Yes, yes, yes. How can we join the offline classes, ma'am? Oh, Shravanable, we are good with online classes here. You don't have to. We are giving you everything here. Sit back home. Don't waste your time going from one place to another and study. Is it clear? Okay, talking about reptilia, guys. Reptilia means reptile, which can creep or crawl. So this is their mode of locomotion. They are mostly terrestrial. Their body is dry and cornified. These were the first true land animals. Fishes completely aquatic, amphibians dual habitat. Now these were the completely and true land animals for the first time. Skin is dry, cornified, epidermal scales and scutes were present. Now what are scales and scutes here? These are scutes. Do you see? Turtles and tortoises, they have scutes. These are scales. On snake also we have scales. That body is called scales. Are these points clear? Now, see, they do not have external ear. Even amphibians don't have external ear, right? Even they don't have external ear. The pinna is not present, yes or no? Please tell me. All clear to everyone? All clear? Right. Then the tympanum represents the ear. So since pinna is not present, so eardrum comes outer on the outer surface. So tympanum represents the ear. This is true for amphibians as well as for reptiles. Then it says there are two pairs. We are talking about tetrapoda. So of course they will have two pairs. Usually three-chambered heart. Fishes have two-chambered and amphibians and reptiles have three-chambered. But there are few reptiles which don't have three-chambered. That is crocodile. Not just crocodile, it is crocodile, it is alligator, it is ghadial or gavialis. All these, they are all crocodile looking, gavialis. Gavialis. They are all crocodile-like structures. They all have four-chambered heart. But just having four-chambered heart does not make it a warm-blooded. They are also cold-blooded because mixing of blood will still take place. If not within the heart, then outside the heart. Now, poikilotherms means cold-blooded. Snakes and lizards shed their scales as skin casts. So they will come out of the body, leave their skin behind, and the new skin will regenerate. This is called skin cast, also known as ecdysis, molting. This is one type of ecdysis. Sexes are separate, fertilization internal. I hope you have made the list. Let me know and write down in the chat or comment later on which are the all phyla, all the 5 to 6 phyla or the taxa which have internal fertilization. You have to write down. All those who are going to write down are going to clear NEET exam with better marks. Is that clear? Gavialis is another species of crocodile. Not species, it's an altogether different genus. Another genus. Then development is direct, so there is no larval stage present. Then talking about, see, these are the examples. This is the turtle or the chelone. Then testudo is the tortoise. Chameleon is the tree lizard. Celotes is the garden lizard. Crocodilus is the crocodile. Alligator is the alligator. See, different genus. Gavialis is a different genus. They both have four-chambered heart, right? Hemidactylus is our common wall lizard. Then poisonous snakes, very important example. So poisonous snakes are naja, that is cobra, Bangarus, which is crate, and vipera is the viper. Is that clear to all? Clear to all? Please tell me. Then we come on to aves or the birds. The presence of feathers. This is a skin modification. Most of them can fly except the flightless birds. What are the flightless birds? It is ostrich, it is penguin, it is kiwi, it is ostrich, penguin and kiwi and few more. So these possess beak. They possess beak. So they don't have teeth. The mouth has been modified into the beak. So beak will help in cutting the food. There are no teeth. The forelimbs are modified into wings. Clear? The body has scales and the hind limbs will have scales, and they are modified for walking, swimming and clasping. That means to hold the tree branch. Skin is dry and without glands. So there's just one gland present, which is the oil gland, also known as the preen gland. Now this is only at the base of the tail. Okay, endoskeleton is fully bony, ossified, but now the bones are hollow with air cavities, and such bones are known as pneumatic bones. Are these points clear? Now the digestive tract of birds have additional chambers called crop and gizzard. Interesting. Now you will see crop and gizzard also found in the cockroach. Now heart is completely four-chambered and they are warm-blooded. Now there will be no mixing of blood, and hence they will be able to regulate their body temperature. They'll be warm-blooded, they'll be homeothermal. We also call them endothermals. Cold-bloodeds are known as ectothermal, they are known as endothermal. Now respiration is by lungs. Now see, flight demands a lot of energy, and energy production demands a lot of oxygen. Is that clear? So basically your lung capacity matters a lot here. Now see, so lungs have special air sacs connected to the lungs to supplement the respiration. So see, usually we see double circulation, but here we are going to see double respiration. So birds show double respiration. Understand this. Sexes are separate. Fertilization again internal, and they are again oviparous, and there is no larva involved, so direct development. Do you see? These are the air sacs, these are the lungs. Is that clear? So this is a PYQ, that crop and gizzard, all birds. Where are they found? In birds and insects. This is a chordate, while insects are non-chordates. Crop will always store the food. Crop is meant for storing the food, be it chordate or non-chordate, and gizzard is meant for crushing the food in both the cases. Acts like a true stomach. Please tell me all these points are clear. Please tell me all these points are clear. Yes, please. Priyanka has written internal fertilization in Porifera, Aschelminthes, Platyhelminthes, then most arthropods, chondrichthyes, reptiles, aves and mammals. Sir, Wasim sir, are you there? Joel, this is Tina ma'am. Sir will be in sir's class, right? Okay. Please tell me all these points are clear. Yes, please. Give me a thumbs up. It's an energy break, and thumbs up gives me energy. Yes, please. So these are the examples: Corvus, Columba, Psittacula, Struthio, Pavo, Echinodermata and Neophron. Neophron is vulture, Echinodermata is penguin. Again, penguin is also a flightless bird. This is flightless, this is flightless. Pavo is our national bird. Hope you know. Is that clear? Yes or no, everyone? And last not the least, we have class Mammalia. Variety of habitats, you will find them everywhere. They live on land, they fly, they live in water. Their most unique feature is the milk producing glands, from where they get their name Mammalia. They have mammae, that is mammary glands, and these mammary glands, they produce milk to nourish their young one. Again, being a tetrapoda, there are two pairs of limbs for walking, running, climbing, burrowing, swimming, flying and so on. Skin has hair. Okay, skin will have hair. Again, not all of them will have. So pinna, external ears are present. So hair may be present at one stage and may not be present at the other, but yes, they will have hair at one or other point of life. Then pinna, present. Different type of teeth, heterodont teeth. Heart is four-chambered, homeothermal, that is warm-blooded, and also known as endothermal. Told you. Respiration through lungs, that is pulmonary respiration. Sexes are separate. Sexes are separate. That means they are unisexual. Fertilization again internal. They are viviparous. They will give birth, with few exceptions which are oviparous, and development is direct. There is no larva involved. Now see the examples here. Oviparous ones are Echidna, that is spiny anteater, and Ornithorhynchus, which is platypus. Rest all the examples are viviparous. It's class going on. Rest all are examples here. Now Macropus, this is a marsupial. That is a pouched mammal. Rest we all see the placental mammals. Rest are the placental mammals. Rest are the placental mammals. Is that clear? Yes, guys, let me know if there is any doubt. Any doubt? Any doubt till here? There will be no break. Dinner, ma'am. Wow. Yeah, even my family wants me to have dinner with them. But now it's time for the study. We can't have a dinner break here. We'll have late dinner tonight. Okay, moving on. Alright, guys. So this is the table given by NCERT. And now see here. Cellular. I have marked the unique features. Cellular only in Porifera. Tissue only in Coelenterata and Ctenophora. Then platy will have basically organ, but organ system in few. But organ system will start from Aschelminthes. Do you see pseudocoelom only in Aschelminthes? Right? Do you see pores and canals only in Porifera? Nidoplast only in Coelenterata. But the table that I am going to give you and share with you, that will be so detailed. You will not have to revise from your NCERT after that. But I want you to read NCERT, attend all the lectures, learn things deeply and by heart, attend all the questions, and then use that table for last minute revision. So I'm going to give you accordingly. Yes? 5-minute break. What are we going to do in that 5-minute break? Yes, guys. Okay, let's talk. Let's have a quick talk. Let's have a water break. Have a sip of water. And let's talk. Tell me, what are your challenges? Tell me what were the challenges that you felt during your NEET exam. The paper wasn't very difficult. There were hardly 7-8 questions which were tricky. It was lengthy. And of course, it's an exam, so that examination fear takes over. What were your personal challenges, which I'm asking you to jot down? Let's share. Yes. 720 was promised to me 3,000 years ago. Wow. King, you're very old then. All right. Time management. Okay. One was time management. All right. Priyanka has written all the names. See anyone? Very good, Priyanka. Very good. I am sure you're going to get better marks this time. Yes, please. Time management, ma'am. Does tympanum represent the internal ear? No, external ear. Tympanum represents the external ear. In our case also, tympanum is a part of external ear. It is not the internal ear. Yes, guys. We face you were hungry during the exam. You should have eaten something. Little panic in the time of examination. That is normal. You know, how to prevent getting panicked? Don't look around much. Have faith in yourself. Whenever you feel panic, deep breathe and think about the good things. That helps me. Think about the good things. Even if you feel panic, you know, think if something has to go wrong, it will go wrong. So I know this is easy to say, but the point is I want to try to feel better. So I have to think something positive. Only positive things can make you feel better. So think about all the positive things, right? 8 questions, silly mistakes due to overconfidence. I am sure, Vishwa, this must have happened. Platyhelminthes is oviparous or oviviviparous? It is oviparous. Remembering, then there was lack of practice. Then that was definitely lack of practice. What else? Filling the OMR sheet. Adila Meran. Okay. Filling the OMR sheet. I'll tell you, there are students who fill OMR at the end and then they tend to miss out the questions. They are not able to fill the OMR properly, and the questions that they have done right, they're not able to answer in the OMR. Always fill OMR in short, parts. You complete one page and you fill the OMR, and your finger should move simultaneously. You have to check the question number and answer, right? So fill it in parts. Don't do it in one go. Paper was tough. Depends. Time management, focusing, hunger, pressure. What else? What else? No, ma'am, time of exam. Invigilators are often asking something. Yes, Monita, I understand, Monita, yeah. But these are the things which everybody is facing. Now, that's where competition comes in. If you can survive in that. Anish, don't you want to become a future doctor? Don't ask anyone. So sharks keep swimming. Won't they get tired? Sharks have a very interesting phenomena. They sleep while swimming. Their brain sleeps in parts. So one part of the brain sleeps while the other is awake, and they keep swimming slowly. And then the other part will sleep and they take turns. So the whole brain doesn't go to sleep in one go. Remaining chapters will you complete today? Yes, Isha. We are going to start with the next chapter now after that. After this, we are going to start with biomolecules. I'm ready. And then structural organization, frog and cockroach. We are going to do that. Girisha, 314 bio is a good score or not? It is a good score. But then, when everybody is getting a good score, how will Girisha stand out? Tell me. So Girisha can stand out if she gets better than 340, isn't it? So why not aim for 360, aim for the sky and you reach the stars. So our aim should be 360. If you're getting 340 out of 360 in bio, you are a good student. But now you have to, give it a little more push. But honestly, this change from 340, this jump from 340 to 360 is difficult. It is difficult. Why it is difficult? Because now there are very minor mistakes that you are making. You know the topic, but you are making silly mistakes, and now this demands change in micro habits, more practice, working on those shortcomings, those few topics that you may not be very thorough about. So identify. Okay, but 340 is definitely a good score. Is that clear? Clear? Girls crying in the exams and disturbing. Vishwanand, you're very unkind, huh? It's not just girls. Boys also cry in exams. Can score above. Ma'am, I got only 20, can score 3. Yes, Shubhalakshmi, you can easily score. This is a great opportunity for those students who want to do better. Right? And now, one thing you should be very sure about, never be scared of NTA, no matter how false proclaims it makes. Remember the proclaims of NTA just before your NEET exam? You don't have to be scared of that. Cockroach is deleted? Since when, this re? It is not deleted. Cockroach is there in syllabus. From zero, how to study to get MBBS seat. Banu. Okay, now I'll take this last message and now we are going to jump on to the chapter of biomolecules after this. If you are at zero, then I would tell you, don't waste this time. Start preparation now. If you feel that, okay, you are at zero and you have to take this Re-NEET, which I'm sure you are not, but even if you are at zero and you have to take this Re-NEET, use this time, take a chance, and whatever you have studied in this 1 month will automatically be of help if you take Re-NEET NEET exam in 2027. So start now. See how much you could study in 1 month, right? But I'm sure you're not at zero if you are a 2026 student, NEET aspirant. All right, guys. Now moving on to I still don't get the last part of this chapter. Study which chapter are you talking about? Ma'am, tomorrow last be at 10 a.m. No, 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 no. Class won't be at 10 a.m. See, I have already informed you, KK sir's uncle has expired, so he won't be able to take the class tomorrow. So we will have another marathon, Zoology Part 2, Class 11, where we will be discussing human physiology. So for next coming 3 days, that is this day is already done. So tomorrow and day after, so Friday and Saturday, we'll be having the marathons. So in these 3 days, we'll be able to complete our class 11 biology. Almost done. Is that clear? This week we'll be completing class 11. All right? Right? All right, guys. Now moving on to the next chapter, biomolecules. Give me a thumbs up. You're all ready. The break is over. I've taken your doubts. I've just talked a few things with you. Let me know. All thumbs up. Yes. All ready for the next chapter with the same energy? I want the same energy back. Biomolecules. So first of all, how to analyze the chemical composition. See, to analyze the chemical composition of any structure, any living tissue, you have to first grind it and grind it in trichloroacetic acid. So this is the solvent that we are going to use. This is the formula. So you have to take a vegetable or any tissue that you want to study in a pestle and mortar and put this trichloroacetic acid. Trichloro. Do you see Cl3? Chloroacetic acid using a pestle and mortar, and a thick slurry is obtained. Now you strain it in a cheesecloth, and whenever we strain, guys, we always get two things. See, we are going to strain this. When we strain, we get two things. One which remains behind is called the retentate, and the one which gets filtered is known as the filtrate. See, whatever is bigger in size will be found in the retentate. So retentate will consist of bio-macromolecules, yes, and the one which gets filtered will consist of the smaller molecules, bio-micromolecules. Also, retentate forms an acid insoluble pool. So this is the acid you have used, trichloroacetic acid. So every molecule that could dissolve in trichloroacetic acid got filtered out. So filtrate has acid soluble pool while retentate has acid insoluble pool. Is it clear? Now micromolecules, what are they consisting of? They are going to consist of this acid soluble pool or filtrate. They have micromolecules, amino acids, nitrogenous bases, simple sugars. Are you getting that? All these monomers, they are found. Vitamins, minerals, they are all found here. Then insoluble pool will have macromolecules, the large compounds, that is macromolecules like proteins, polysaccharides. Then you will have nucleic acids. Is that clear? Now fats are micro, but they will be found in the retentate. Why? We'll talk about this. Please tell me these points are clear, right? So see, retentate will have bio-macromolecules plus fat. Fat is actually a micromolecule, and macromolecules consist of polymers like proteins. This is what I've shared already. And then micro will have monomers of these protein monomers, amino acids, the monomers of nucleic acid, nitrogenous bases, then sugars, vitamins, minerals. Now how do we, if I want to study about minerals, that which mineral is present, we have a destructive method. So in order to particularly study minerals, we are going to follow the destructive method. What do? We burn the tissue. What do? We burn the tissue, guys. So what will happen? When the tissue is fully burnt, carbon compounds will be oxidized to form carbon dioxide and water vapors will be removed. All you will be left behind is ash. Now this ash contains the minerals, calcium, magnesium, etc. So inorganic compounds like sulfate and this, they are seen in the acid soluble pool. Are these points clear? Easy. Now moving on to the next. See, inorganic compounds and organic compounds. See, but we should classify these bases and fatty acids and amino acids. Now let's discuss them one by one. Let's talk about these biomolecules one by one. Yes, please. I'm going to start with bio-micromolecules. The first micromolecule is the amino acid. Why do we call it amino acid? Because it has two groups. It has amino group as well as it has the acidic group. So it is an organic compound with amino group and acidic group, and we can call it a substituted methane. See, this is the formula for methane, CH4, right? Now in methane, we are going to replace these 3 hydrogens, one with amino group, another with carboxyl group, and the third with a variable R group, and we have one H. Now such carbon which has all the 4 different groups, such carbon is known as alpha carbon or chiral carbon. Such carbon is known as chiral carbon. Now in this world, in this living world, the amino acids that are present, all the amino acids will have these 3 groups. These 3 groups are constant in all. Now the difference between all the amino acid lies here. The whole difference lies in this one R group, and this R group is going to produce around 300, 400 different types of amino acids. Is that clear? Clear to all? Now the type, based on the nature of R group, we have different types of amino acids. Is that clear? Now out of these 200 to 400 amino acids that I'm talking about, only 20, what is written? Only 20 amino acids are going to help in protein synthesis. Is that clear? Only 20 amino acids will help in protein formation or protein synthesis. Not all of them will form proteins. Are these points clear to all? Please tell me. Please tell me. Right? Now these are the 2 tables given in biomolecules. You make sure that you know them by their, not all of them, at least the top 4. See, in Earth's crust, it is the oxygen. See here. See, in Earth's crust, oxygen 46.6. That is the maximum. Then silicon 27.7, calcium 3.6, and sodium 2.8. While in human body, do you see we have these values? So you should be able to at least mark them in the decreasing order, and the order has been written here. Please tell me that these points are clear to you all. Yes guys, all clear? Please let me know. And don't forget to hit the like button guys, and the bell icon also, so that you are notified. And in case guys you are liking these and you want to attend all these, please don't forget to join me tomorrow also and day after also at 7 p.m. Our time will be 7 p.m. And in this week we are going to complete our class 11 biology. Tomorrow I'll be discussing human physiology, so it is very, very important. Is that clear? We'll be discussing NCERT, we'll be discussing all the topics in detail, and you can see it, we are discussing it from 0 to hero level. Right? Now this is not any important. Now we were talking about amino acids. Now I told you it is the R group which is going to produce the different varieties. So R group in the proteinaceous amino acids could be hydrogen, because it can be hydrogen. That means, see here, see. So these are the proteinaceous amino acids. Proteinaceous amino acids means these amino acids will participate in protein synthesis. Now these 3 groups, I told you, these 3 groups are constant. These 3 groups are constant in all the amino acids. The difference lies here. If R group is H, R group is equal to H, this amino acid is called glycine, and this is known as the simplest amino acid. Then see, if the R group is CH3, that is the methyl group, then amino acid is known as alanine. If it is serine, then the R group will be equal to CH2OH. Now this OH makes it alcoholic. So serine is an example of alcoholic group. While see, glycine and alanine are both neutral amino acids. They are neutral amino acids. Do we see that? Do we see that here? So 20 amino acids are going to help in protein synthesis. Please tell me that these points are clear. Now, other than these, see, the chemical and the physical properties depend on amino, carboxyl and R functional group. Number of amino and carboxylic groups are going to make it acidic. Now see, amino group is basic in nature. Carboxylic group is acidic in nature. So in one amino acid which has one amino and one carboxyl group, so it should be balanced. This is acidic, this is basic. So overall it should be neutral. But what if this R group has another NH2? What will happen? So sorry, this is acidic, This is basic, right? So what will happen if there is another amino group in the R group? This is also basic. So overall basic nature of amino acid will dominate, yes or no? One acid, one base, sorry, one basic, one acid group, they will neutralize. But when there is another basic group here in the R group, the overall nature of the amino group will, amino acid will become basic in nature. Yes or no? So this is what it say. So you have to remember that there is acidic amino acid called glutamic acid or aspartic acid. The name itself says the nature. Then lysine. Lysine is basic in nature. Can you name any other? Can you name any other basic amino acid? One is lysine, another is arginine. And do you remember both of them are found in histone proteins? Yes or no? And that is what make histone proteins basic in nature. Please tell me if you remember this. And neutral, the example is valine. And then there are two more examples of neutral amino acids that I have shared with you. Why? Because, yes guys, which are the two other examples which I have shared with you? Quickly let me know. Arginine, histidine, yes. Alanine, right? And I have told you about glycine. Is that clear? Now, in case the R group has a benzene ring, then there are three examples: tryptophan, phenylalanine, and tyrosine. These are the examples of aromatic. Aromatic means they have that ring structure. And then, okay, so that's it. Now, talking about the particular property of amino acid is the ionizable nature. So I've just told you, see, this amino group is basic, okay? This acidic group is acidic. Both can get ionized, yes, both are ionizable. So, they will be forming different ions in different pH, right? If you change the pH of the solution, a different ion will be produced. Now, it is a very, very characteristic feature. See here. So, this is something we have already shared. So, this is an amino group, basic, and it forms a positive radical. See here. While this is the acidic group, carboxyl group, and it forms a negative radical. Yes? Okay. Now, this is the repetition, sorry. Okay, now I want to discuss this. Now, in a particular solution, see, this amino group can get ionized and it can form NH3 positive. It can form NH3 positive. While in a different solution, this carboxyl group can get ionized and it can form an acidic radical. Yes? While in the state of equilibrium, both amino and carboxyl can form an ion simultaneously. They can both undergo ionization simultaneously. So there will be a positive charge and a negative charge which will coexist. Now this is a unique property of only amino acids, that they can form an ion which has both the charges existing simultaneously. Now such hybrid ion is known as Zwitter ion. Zwitter means hybrid. It is a word from German language. So it is a characteristic feature of amino acid. Please tell me that these points are clear to you all. Yes, please. Please tell me these points are clear to you all. Yes, the Shirisha, what is the doubt, bache? What is the doubt? How to continue up to Re-NEET? Joel's, please get in touch with the tech team. Please get in touch. Your subscription will be enhanced. Yeah, it will be, it will continue till your NEET exam. Please teach genetics like this too, with NCERT coverage. Every chapter will be covered like this. Don't worry about that. Is that clear? Every chapter will be covered like this. All right, moving on now to the next, that is lipids. So another micromolecule that we are going to talk about is lipid. So already told you, lipid is a micromolecule, but it will be found with the retentates. It will be found amongst other macromolecules. Why? Because see, when we are grinding the tissue in a pestle and mortar by adding trichloroacetic acid, what happens? The membranes are breaking. And we have lots of lipids in our membranes, bilipid layer, remember, plasma membrane. So when we are crushing the tissue, the plasma membrane is breaking down into small vesicles. We are still not able to, dissociate each and every lipid separately. One lipid is micro in nature, but what we get is not one lipid. We are getting group of lipids in the form of vesicles. Now, when so many lipids are together, they form a vesicle. Now, vesicle has a larger structure, larger size, which is in accordance with the size of macromolecules. Do you understand that? Do you understand that? Okay. Now, talking about lipids then, water insoluble. We always see oil and ghee floating on water because they are insoluble. Now, they could be simple fatty acids with a carboxyl group attached. Why do we call it a fatty acid? Again, for the same reason, it has a carboxyl group attached to the R group. Now, R group can be methyl or ethyl or higher number, and the number can be from 1 to 19 carbons. For example, palmitic acid will have 16 carbons, including carboxyl group. Now, point to be noticed here. When we say palmitic acid, now remember, palmitic acid has how many? Palmitic acid has C16. Palmitic acid has C16, but this 16 will include which one? So basically it is C15 and COOH. So that's how it becomes C16. So that is why it is written including carboxyl carbon. Now in case it says number of carbon atoms in palmitic acid excluding, what will be the answer? Let me know in the chat. Let me see if you are listening properly. Yes guys, what will be the number of carbon atoms in palmitic acid excluding carboxyl carbon? Please let me know. Very good, Vidara. Very good. Vidara is first to answer. 15. Is that clear? So that's how you read the question properly. Now, similarly, arachidonic acid will have 20, again, including the carboxyl carbon. So, if it says excluding, then again it will be C19. Is that clear? Now, fatty acids can be saturated or unsaturated. Now, I want to share these things. See, let's discuss this. So this is fatty acid. How is fat formed? Fat is formed with one fatty acid and glycerol. Do you see here? So it is R group and COOH, and it has written R group can be anything, and the number of R group can be between, it will lie between this R group. It can be 1 to 19, right? And then we have the COOH group. Is that clear? Okay. Now this is one fatty acid. Now there is glycerol. Please see, glycerol is one of the simplest alcohols with 3 carbons, CH3OH, CHOH, CH3OH. It has 3 carbon. It is 3 carbon alcohol. So what is the chemical name for glycerol? Trihydroxypropane. Clear to all? Now one glycerol molecule will always be 1. Remember that. This will be always 1. Fatty acids can be 1, 2, 3. It can be 1, 2 or 3. Is that clear? Now in glycerol molecule, do you see there are this OH, OH, OH? Now these hydrogens are replaceable. If you replace one hydrogen with one fatty acid, you will form this ester bond. So what are the bonds formed in lipids? So the bond formed between glycerol and fatty acid is the ester bond. Is that clear? Is that clear? So this is one glycerol getting esterified. Esterified means forming ester bonds with 3 fatty acids. Now this is a true fat. This is a true fat. It is not necessary that it will form a true fat, means all the 3 hydrogens will be replaceable. Now see here. So this is something we have already discussed. See. Now see, glycerol, one molecule with one fatty acid, it will remove one water molecule and form one ester bond, and such fat is known as monoglyceride. If there are 2 fatty acids, 2 water molecules will be lost, it will form diglyceride. Then 3 fatty acid, 3 water molecules, triglyceride, and this is a true fat, this is a PYQ. So number of ester bonds is equal to the number of fatty acids attached and the number of water molecules lost. Are these points clear to all? Please tell me. Please tell me these points are clear to everyone. Is that clear? Can we move on? Now see, your fats can be saturated or unsaturated. Saturated means they will only have single bonds, and unsaturated means presence of at least one double bond or more. Now fatty acids saturated ones, they will only have single bonds. We call them animal fat like butter, ghee, margarine, and they are solid at the room temperature, and they are known to increase the cholesterol level, है ना? Increase the cholesterol. While saturated fats are commonly known as oils, they have one or more double bonds. They are always plant based, so oils, and they are liquid at room temperature, except coconut oil, because coconut one is a saturated. That is an exception. Is that clear? Are these points clear to all? Please let me know that these points are super clear. Right? Now moving on to, now see, other than fatty acid and glycerol, there can be a third element. Okay? Now the third element could be anything. For example, in phospholipids, see, so this is the palmitic acid. Let's see all these. So this is 1 COH and 14 and 15. So in total, C16. Palmitic acid. Do you see the glycerol? This is the trihydroxypropane. This is a typical structure of triglyceride, while this is CHO, OO. All the H have been replaced with the different fatty acids. This is a typical triglyceride. Now I am going to talk about when the third element comes in. Talking about phospholipids. What will be attached? Phosphoric acid. See here, this is the glycerol. This is the glycerol, right? Now one side we have fatty acid. This is fatty acid. This is fatty acid. And here comes the phosphoric acid. Yes or no? And then we have another group here. So because there is phosphoric group attached here, we call it a phospholipid. Now one very important example of phospholipid is lecithin. Very important example. We have lecithin on the inner side of our alveoli also, found in the cell membrane. See, lecithin is one example. Some tissues like neural tissues have lipids. With more complex tissues, there are difference. We don't have to talk about it. Lecithin is found on this is alveoli. This is where lecithin is present. If lecithin is not present, the alveolar membrane will degenerate. It will degrade. Is that clear? So these are the examples of phospholipids. Please tell me these points are clear. Please tell me these points are clear. Now we are going to talk about another micromolecule, which is nitrogenous bases. Now see, nitrogenous bases are heterocyclic rings. This is one component of your DNA or RNA of your nucleic acid. See, we know nucleic acids are made up of nitrogenous bases plus sugar, pentose sugar, plus what? Phosphoric acid, right? Now nitrogenous bases are further of two types, purines and pyrimidines. Now purines, as I've told you, purines have double ring structure. Purines have a double ring structure, while pyrimidines have a single ring structure. So these are 6-membered, these are 9-membered. Is that clear? Now see here, I want you to see this, right? So pyrimidines are cytosine, thymine and uracil. You remember cut, single ring, 9-membered, double ring and 9-membered. These are purines, adenine and guanine. Clear? Now thymine will only be found in DNA. We know that. Uracil will only be found in RNA. Rest 3, cytosine, adenine, guanine, they are common to DNA and RNA. Now we know the structure of nucleic acid. There is a pentose sugar present, that means C5 sugar. Now ribose is present in RNA, while deoxyribose is found in DNA. Do you know the difference between the two sugars? Do you know the difference? Let's see. So this is the structure of ribose. This is carbon 1, 2. In 2 we have H and OH. This is ribose sugar. And what will be the difference? We will minus 1 O and it will become a deoxyribose sugar. At carbon number 2, we will minus oxygen at C2 and it will become a deoxyribose sugar. Now, this is going to make, this is O, this is H. So, because of the OH, it is very reactive. That is why RNA is less stable, because of this OH. Is that clear? Is that clear to all? Now, another very important thing. See, nitrogenous base, all the nitrogenous bases, added with sugars, deoxyribose or ribose, they are going to form a nucleoside. They are going to form a nucleoside. While if I attach the third element, that is phosphoric acid, it is going to form a nucleotide. And nucleotide is a monomer of nucleotide. nucleic acid. So see, this is a sugar, this is a nitrogenous base. Sugar and nitrogenous base, they will form a nucleoside. Now in a nucleoside, if I add phosphate, this will become a nucleotide. Please tell me these points are clear to you. Please tell me these points are clear to you. Yes guys, please tell me. Quick, quick, quick. I'll pin the name of the I'll pin the Telegram channel in the description box, guys. Don't worry about that. All clear? Perfect. Okay, moving on then. Moving on. Now see, you should be able to identify the names. So these are the names of these are the names of nitrogenous bases: adenine, guanine, cytosine, thymine, uracil. When you add sugar molecule to it, it becomes a nucleoside. So adenosine, guanosine, cytidine, thymidine, uracil. You will get a question, which of the following which of the following is a correct set of nucleosides? So you should know the difference between adenine and adenosine. Adenine is individual nitrogenous base, while adenosine will have a sugar attached to adenine. Are you getting that? Similarly all. Now, if I attach phosphate to adenosine, it will become adenosine monophosphate. If I attach this, guanosine monophosphate, cytidine monophosphate, and so on. Is that clear? But other than adeno monophosphate, because one phosphate is getting added, it is phosphoric acid. So can I call it adenilic acid? Can I call it guanilic acid, cytidilic acid, thymidilic acid, and uridilic acid? So these names represent the nucleotide, these names represent the nucleoside, and these are the names of the nitrogenous bases. Please tell me these points are clear to you. And now these are the structures given in your NCERT. Do you see the structure of adenine? It represents a purine, 9-membered double ring structure, 6-membered single ring uracil. Now see, adenine, this structure added to the sugar, this becomes adenosine. And when phosphoric acid is attached to adenine and sugar, it becomes adenilic acid. It is a nucleotide. Please tell me that these points are clear. While this is a nucleoside, do you see? And these are the nitrogenous bases. Similarly, uridine, you have sugar attached to the uracil. And if you attach phosphoric acid here, it becomes uridilic acid. Please tell me these points are clear. Yes, please. Please tell me these points are clear. Quick, quick, quick. Yes, guys. Surya, see, you click on the link and you scan the given, you scan it and you can easily watch. If there is any issue, you can always contact the tech team, but it should happen. Just restart and you will be able to see. Is that clear? All right. Now, moving on to, see, this is the ribose sugar that I was talking about. This is the RNA sugar. Add carbon 2, you minus oxygen and it becomes DNA sugar. This is the structure of glucose. Clear? Now, moving on to the next, this is very, very important. Primary metabolites and secondary metabolites. What are metabolites? First of all, metabolite means anything produced out of metabolism is a metabolite. Now, primary metabolites means your body cannot do without them. Your body cannot survive. They are ultimately necessary for the survivor. They are so important. Proteins, carbohydrates, fats, they are all your primary metabolites. They are required for the normal physiological activities. So, these are primary. However, if you analyze plants, animals, and microbes, then plants, animals, and fungus, microbes, plants, fungi, and microbes, other than primary metabolites, they produce some secondary metabolites also. Animals don't produce the secondary metabolites. These are primary. Is it clear? However, when one analyzes this, one would see thousands of compounds other than these primary, and all these compounds are known as secondary metabolites. Now remember, animals will never produce secondary metabolites. Is that clear? Animals don't produce that. Who produces secondary? Plants, fungi and microbes. Is that clear? Now what are secondary metabolites? See, written primary metabolites have physiological processes while secondary metabolites are not used by the host organism. Is that clear? We do not at the moment understand the role of all the secondary metabolites in the host organisms. So which are the hosts? Plant, fungi, microbes. Those who are producing secondary metabolites are not using them themselves. So they are useful to the humans, for example, rubber, drugs, spices, scents, pigments, and they have ecological importance. So these are the two functions of secondary metabolites: human welfare and ecological importance. And then this table comes in. This year in NEET 2026, there was no question from this table. But let's wait for Re-NEET, because usually in almost every year we have got question from this table. So get prepared. So alkaloids, morphine and codeine. So you should know which chemical belongs to which category. So you should know the category of the chemical. Carotenoids, anthocyanins, these are the pigments. Terpenoids, see, they're easy to remember. Terpene, terpenoids. Oils. Now the ones which I have put in the bracts, right, these will require an extra effort. For example, morphine and codeine, these are alkaloids. Aspirin and ricin, these are toxins, very fatal toxins. Concanvalin is a lectin, while drugs are vinblastin and curcumin. Curcumin comes from haldi, turmeric, and vinblastin is from Vinca rosea, Sadabahar, which is used for leukemia treatment. And polymeric substances are rubber, gums and cellulose. Now they come in the form of match the following. Please tell me these points are clear. Yes, please. Yes, please. All clear? Moving on now to the next, that is biomacromolecules. So we have read biomicromolecules. So what are the differences? How do we categorize or classify the molecule into micro and macro categories? See here. Micromolecules will have size between, or the weight between 18 to 800 Daltons. That's it. And that's why we put fats also in the category of micromolecules, because the weight doesn't exceed 800 Daltons. But still we read them, we find them in retentate, because I've told you individual lipid is a micromolecule, but when you crush the tissue, what you get is not an individual lipid, you get vesicles. So vesicles have a larger size, okay, but as an individual lipid, it has size less than 800 Dalton. So it is a micromolecule. But the retentate, that is the macromolecules, they are found in acid insoluble pool. So macromolecules consist of proteins, nucleic acid, that means all the polymers. Can you see polysaccharides, not the lipids here? Okay, so they have just written about insoluble pool. So insoluble pool has this, but it is not a macromolecule. That's why I put across. Anyway, so the molecular weight is in the range of 10,000 Daltons. See, it is written with the exception of lipids, rest all the three, proteins, nucleic acids and polysaccharides, because these are polymers. They have size in the range of 10,000 Daltons or above. So we differentiate it on the basis of molecular weight. Micro will have between 10 to 800 Daltons and macro will have in the range of 10,000 Daltons or above. Is that clear? Okay. Now moving on to, see, this is, I've already explained why lipid is found in the retentate, because we get them in the form of vesicles. Now acid soluble pool. Acid soluble pool represents the cytoplasmic composition, that is the micromolecules, biomicromolecules. This is the soluble and the cytoplasmic composition, while macromolecules form the cytoplasm and organelles become the insoluble pool. From cytoplasm and organelles form the acid insoluble pool. These lines should be learned as such. Clear-cut line. It can be replicated in your NEET exam, yes or no? It can be replicated, yes or no? Please let me know. So guys, please stop talking to each other, talk to me, or just revise? Hi Ansh. Table chart for this lesson, Animal Kingdom. Okay, Dhanush, you will get it. Can't wait for human physiology. So can't I. Yeah. Alright then, together they represent the entire chemical composition of living tissues or organisms. Are these points clear? Now, another very important table. Average composition. You get this question and match the following. Do you see that the maximum percentage of cellular mass is that of water? Right? Water is 70 to 90%. Then proteins 10 to 15%. Carbohydrates 3%. Lipids 2%. Nucleic acids 5 to 7%. Ions 1%. So what is the decreasing order? What is the decreasing order? Water, then proteins, nucleic acid, carbohydrates, lipids, and ions. How do I remember it? Water is of course the maximum. How do I remember? I remember pinacally. This is my way of remembering. Pinacally. Now, if you remember the decreasing order, can you automatically put them against match the following? Yes. But you should know the data also in case they just give one line that nucleic acids have they are present in 5 to 7% in the cytoplasm, then that would be right. Not the cytoplasm, sorry, in the cell. Is that clear? Now, talking about the macromolecules, we have discussed, we have seen the percentage, we have seen their fraction in the cell. Now, talking about proteins, we call proteins as polypeptides. Proteins are the polymers of amino acids, and amino acids are going to join together through peptide bonds. So since there are multiple peptide bonds, we call them polypeptides. It is a linear chain of amino acids linked by peptide bond, polymer of amino acids. And how many amino acids, different type of amino acids can participate? It is only 20: alanine, cysteine, proline, tryptophan, and lysine. Yes, since different amino acids are going to join together, we call it a heteropolymer and not a homopolymer. Is that clear? Now, there are two types of amino acids. The one which we take through our diet. See, it is important to eat pulses or eggs or non-veg if you are non-vegetarian then. So, what are we getting? We are getting proteins. Those proteins are broken down to form amino acids. Why it is so important to take those proteins? Because our body cannot produce it. So these are called dietary proteins. They are essential. Essential means they have to be supplied through our diet. Body cannot produce them. While there are others which are non-essential. Why non-essential? Because you don't have to take them. It is not essential to take them in your diet because our body can make them. Is that clear? Is that clear? Now, what are the functions? Some functions which are carried out like transport of nutrients, fight against infections, some are hormones and enzymes. So, there is this table. Very important table. Again, you get many questions from this table. So, collagen. Collagen is intercellular ground substance, most abundant protein in the animal world. Trypsin is an enzyme and it is protein digesting. Insulin is a hormone which regulates your glucose level. Antibodies fight the infections. Receptors are for sensory reception, and GLUT4 helps in the transport of glucose into the cell. So GLUT means glucose transport 4. So we have different types of receptor molecules. GLUT4 is one of them. Please tell me these points are clear. Please tell me these points are clear. Vishwa, which type of secondary metabolite are you talking about? We have similar things. For example, if we talk about opioids, our body also produce opioid-like compounds, are natural painkillers. So that is why we have some receptors for opioids. I hope it answers your question. Do we have to remember? No, Smicky. We don't have to learn the examples of essential and non-essential amino acids. We don't have to. Now, moving on to the next. So, this was 2024 question as such. Is that clear? Now, collagen is the most abundant and then rubisco is the most abundant in the whole biosphere. Collagen in the animal world and rubisco in the biosphere. Are these points clear? Easy. Now talking about the polysaccharides. Polysaccharide is again a macromolecule made up of so many small sugars. Again, acid insoluble pool, right? Long chains of sugars. For example, cotton thread. So cotton that we use is a polysaccharide and it represents cellulose. So see, glucose is the monomer, and from this one monomer we can form many polysaccharides. It depends how we join them, how many number of glucose molecules are present. So glycogen, starch, cellulose, they are all the polymers of glucose. Then from fructose we form inulin. Inulin is found in the dahlia tubers and it is used in the kidney test, kidney functioning test. Is that clear? Is that clear? Now see, all these have one monomer. So these are all are homopolymers. These all are homopolymers. So starch is the stored form of sugar in animals. This is also known as animal starch. Starch is a stored form of food in plants. Yes. Cellulose. Cellulose is an example of, your paper is made up of cellulose. Cotton, that is all made up of cellulose. Is that clear? Now very important lines here. Now see. Very important example lines here. The right end of the reducing end, right end of the chain is the reducing. To remember, RR. So right end is going to be the reducing end and left end is going to be the non-reducing end. So when you are joining glucose, 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 so towards my left hand side is the non-reducing, right hand side is the reducing side. Is that clear? So that's what you have to remember. Now we know about the starch test. We all have performed the starch test. How do we check it? We put a drop of iodine. If it turns blue-black, it means starch is present. If it maintains its color, that is yellowish-brown, then starch is not present. So why that starch gives test with iodine? Why starch gives test with iodine? See, starch forms helical secondary structure and it can hold iodine. So the helical structure of starch can hold iodine and this gives the blue color. Now we don't get a similar test or a blue color in case of cellulose. Why? Cellulose does not contain the helices. When it doesn't have a helical structure, it will not capture iodine, it will not give color. So no color change in case of cellulose because of no helix. No helix here. And helical structure is found in starch. Helical structure is also found in glycogen. So starch gives color with glycogen also, just a different color, violet red color. Please tell me these points are clear. Do you see NEET 2023 question? Cellulose doesn't form blue color with iodine. Why? It doesn't form a helix and does not contain iodine. Please tell me all these points are clear. Now see, this is the structure of glycogen I was talking about. So this is the left hand side and hence this is the non-reducing and this is the right hand side, so reducing. Clear to all? Now plant cell walls are made up of cellulose, paper, cotton fiber, all cellulose. So I've told you. Now there is another type of polysaccharides which is called complex polysaccharides. Complex polysaccharides will have different types of monomers. For example, now see, for example, glucosamine, N-acetyl-galactosamine, chitin. Chitin is a complex polysaccharide. It is a homopolysaccharide. Chitin is a homopolysaccharide. And what is the monomer compound of chitin? It is NAG, N-acetyl-glucosamine. Yeah. So this is galactosamine. Similarly, we have N-acetyl-glucosamine. Please tell me these points are clear. Is this point clear to all? Please tell me. Quick, quick, quick. Anyone with any doubt? Anyone with any doubt? Please let me know. All right. Photosynthesis and respiration when? Photoya, that will be on Saturday. Plant botany we are starting on Saturday. Now, nucleic acid, again a polymer, told you, polymer of nucleotides. So it will be found in the retentate insoluble pool. And which are the building blocks, that is the monomer of nucleic acid, which is nucleotide. We have discussed the whole thing in detail already, nucleotides. Now, talking about very important structure of proteins. So, we have 4 different structures. Primary structure. Primary structure is always a linear structure. So, it is as if you have opened the protein in the form of a line, and you can easily see which is the first amino acid and which is the last amino acid. So, if you have to count the number of amino acids, you have to see the arrangement, you go for the primary structure. So, when a protein is formed in the initial basis, that immature protein has primary structure. Every has a primary structure. Is that clear? See, structures. Immature proteins will have primary structure. Primary means 1D. Linear structure, number, you get an idea about number of amino acids and order of amino acids, like that. Then we have 2-dimensional structure or secondary structure, alpha helix, and it is a right-handed helix all the time. It is beta pleated or beta pleated. So the structure of keratin, that is the one which is protein found in a hair and nails, that has alpha helical structure, while the silk protein structure has a beta pleated. Silk fabric that we are talking about, which is secreted by a silkworm, it has beta pleated structure. Now, tertiary structure or 3-dimensional structure is like a ball of wool. That means you just wind it. It is a 3D structure. Now, this 3D structure is very important for biological activity, for example, in case of enzymes. And then we have a quaternary structure. This quaternary structure is present when a protein has more than one polypeptide chain. See, when a protein is made up of more than one chain, for example, hemoglobin, it has 2 alpha and 2 beta. So there are 4 chains. Such proteins will have quaternary structure. So where every chain will have its own primary, secondary and tertiary, but together when it comes together. So when all the 4 chains come together, that whole substructure will be having quaternary structure. See, hemoglobin, antibodies. In antibodies also there are 2 heavy chains and 2 light chains. So they also have a quaternary structure. Please tell me that these points are clear. Chitin is a homopolymer. Yes, yes, yes. Is that clear? Okay. Now moving on to, see, these are the quaternary structures I have discussed. So this is the structure given in NCERT. So this is the linear structure, primary. This is the helical structure, alpha helix. This is in sheets, beta pleated. This is just a 3D structure, tertiary. And this represents a quaternary structure. Is that clear? Is that clear? So I have discussed the structures of proteins, and now last not the least, one of the very important parts of this chapter is enzymes. Coming on to tertiary structure. What is the structure of enzymes? Just read, tertiary structure, important for the biological activity. Another important line, almost all enzymes are proteins. It is not saying all enzymes are proteins. If we don't add the word almost, the statement goes wrong. Why? Because we do have exception of ribozyme, which is a RNA, Now, enzymes, like any protein, will have secondary and tertiary structure. If we open them up, they will have their primary structure also, secondary also. But they function like enzymes only in the tertiary structure. Are these points clear? Are these points clear? So, these enzymes will have a particular site where the substrate binds, and that site is known as the active site. Now see, enzymes get damaged at high temperature. So they work at a very specific temperature, okay, not above 40 degrees Celsius. Enzymes isolated from the organisms living under extremely high conditions. Yes, that is also possible. One example is of Taq polymerase, which is obtained from Thermus aquaticus, which is used during, Taq polymerase is used during PCR, polymerase chain reaction. I hope you remember. How many of you remember this PCR? And now this Taq polymerase is stable even at a very high temperature of up to 80 to 90 degrees Celsius. Are these points clear? And this is obtained from a thermophilic organisms. They are arc bacteria, of course. Is that clear? Now, talking about how does these enzymes function? See, how does these enzymes function? These enzymes are going to increase the rate of reaction. These are going to increase the rate of reaction. One such example is of carbonic anhydrase, fastest enzyme. Now, what reaction does it catalyze? It catalyzes the addition of, reaction between CO2 and water. Where is it present? Majorly in RBC, a little amount also in plasma. Is that clear? Now, see, it is so fast that in 1 minute, it can produce 36 million molecules. So this is the ability of an enzyme. Now see, the general rule is that rate doubles or decreases by half for every 10 degrees Celsius. So for example, if the temperature is 0 and you bring it to 10 degrees Celsius, the rate of reaction will also, the enzyme, that will also double up. Is that clear? You increase another 10 degrees Celsius, it will again double up. And then it will fall also on the same pattern. Are these points clear? So this is Q10 law. This is known as Q10 law. So this I have already discussed with you about this. So all the reactions, see, now remember, all the metabolic reactions in our body are enzyme catalyzed. There's not even a single step which is not enzyme catalyzed. And we talk about those enzymes during glycolysis, Krebs cycle and so on. We'll read more about that. Is that clear? We'll read more about that. Now, moving on to another, how do enzymes bring about such chemical conversion? So I've told you there is an active site where substrate is going to bind, and when this substrate will bind to the active site, see here. So enzyme will bind to the substrate will bind to the enzyme on that active site, and it will form enzyme substrate complex. You should know this in the sequence. And then once substrate is attached to the enzyme, it will change into the product. So we will have enzyme product complex. And then product will separate from the enzyme, and this enzyme can be reused. This is what explains why we need enzyme in the small amount, because the enzyme can be reused. Are these points clear? Now very important graph. See here. On the y-axis, guys, we take energy, potential energy, and on the x-axis, we are going to take the progress of the reaction. This is the energy of the substrate, ES, and here we see the energy of the product. Now, what happens? You always supply some energy in the beginning, and that energy will take that substrate to the transition state. Okay. Now, this energy which takes it to the transition state is known as activation energy. And after transition state, transition means after which change will start taking place and substrate will start becoming a product. Now, this is a very temporary stage. It is unstable. It will soon start becoming a product after this. And you see, all that energy is being lost and it is changing into the product. So what is activation energy? Activation energy is the energy product of the transition state minus energy of the substrate molecule. Do you see that difference between energy of the transition state and energy of the substrate molecule? Please tell me that these points are clear. Please tell me these points are clear. Yes or no? Right? Now, what is the function of enzyme? Enzyme is basically going to decrease this need of energy. So, the energy which was this high to convert the substrate into product, now in the presence of enzyme, we only need this part of the energy. So, what is the main role of enzymes? Enzymes, they lower the activation energy. So, the transition state which reached that far, it will occur quickly. Are you getting that? So, it will occur quickly. Is that point clear to all? Now, from this figure, we can easily detect whether it is an exothermic reaction or an endothermic reaction. Now, see. Also, before that I must tell you, see, we get this for labeling, that this arrowhead represents the activation energy without enzyme and this smaller one represents with enzyme. Second, whether we use enzyme or we don't, the energy of substrate and product, that will not change. So, initial and final energy will always remain the same, be it in the presence or in the absence of enzyme. Are these points clear? Second, now from this diagram, we can easily tell whether it is exothermic or endothermic. See, initial energy is high and final energy is low. it means it must have lost some energy. So whenever guys ES is more than EP, it is always an exothermic reaction. So this graph represents an exothermic reaction. Now in case this product would have reached here, so like it would have been here. So do you understand? If EP is greater than ES, in that case it would always be an endothermic reaction. Easy? Please tell me these points are clear to you all. Please tell me these points are clear to you all, and I have just explained this point to you. Just explained. So we have discussed this. Now these points are very important. What is the correct order in which enzymatic action takes place? So see, told you first enzyme and substrate will bind to the active site. Now binding of substrate will induce the enzyme to change its shape so that it fits tightly into it. See, so this is substrate binding to the enzyme, and then this was the shape of the active site. But when the substrate binds, it changes the shape of the active site so that it fits more tightly. Now after this what will happen? In the active site, now the substrate will change into the product. It will break the bonds and form new bonds and new product. So this is enzyme product complex, right? And then product will be lost and this space will be available for the new enzyme. So you should know these steps in order. NTA is loving these sequences which it has given clearly in NCERT. Is that clear? Is that clear? So we will be also revising all these sequences during our lectures, and I'll take a separate class also. So see, this was a question in NEET 2024, correct sequential steps. Already the question has come up. Please tell me these points are clear. Please tell me these points are clear. Anyone? Please tell me. All right. Now, moving on to factors affecting. Now see, there are many factors. First of all, it is pH. Every enzyme will have a fixed pH at which it works the best. So this pH where it works the maximum, which shows the maximum. So on y-axis, we take the enzymatic activity. This is the maximum activity. It is seen at a particular pH, and this pH is called the optimum pH. Okay, and similarly, we see maximum enzyme activity at a particular temperature. That temperature will also be known as T optimum. Is that clear? Now for us, it is usually 37 to 40 degrees Celsius. Now, below the pH, optimum pH, or above the optimum pH, the activity is going to decline. Similarly, below the optimum temperature or above the optimum temperature, activity is going to decline. Is this point clear? Is this point clear? So what type of graph do you get here for temperature versus enzyme activity? It is bell-shaped, and the same is for the pH, bell-shaped graph. Are these points clear to all? Please tell me. And now, coming on to concentration of substrate. Now see this. See here. Okay. Now see, on the x-axis, guys, we take S in brackets. That means substrate concentration. And on the y-axis, we take the velocity, Vmax. Is it clear? And when we draw, we get a rectangular hyperbola. So that means initially, if you increase the substrate concentration, velocity is also increasing. But at one point of time, beyond this step, we see if we increase the substrate concentration, the velocity does not increase, rather it becomes parallel. It becomes straight to the substrate concentration. This shape is known as rectangular hyperbola. Now, why doesn't it change after a certain level? The point is this. Now see, for that to understand, we have taken so many enzymes and we start adding substrate molecules. These pink ones are the substrate molecules. So we have a limited amount of enzyme here, right? So in the beginning, when we keep adding substrates, these substrate molecules are getting added to the enzymes. So as we are increasing the substrate, enzymes are getting attached and the activity is increasing. But then at one point, this one point, what will happen? All the enzymes are occupied. We don't have any more vacant enzymes to function. So even though if we increase the substrate concentration, the velocity will not further increase. So the maximum velocity that we obtain is called the Vmax. Now corresponding to this, this is the Vmax, half of Vmax. This is the half of Vmax, right? Now half of Vmax, if plotted on the graph, the corresponding concentration of substrate concentration is known as Km. So what is Km? It is Michaelis-Menten constant. What does it represent? It is the substrate concentration at which half of Vmax is obtained. Please let me know that this point is clear to you all. Yes guys, please let me know this point is clear to you all. Is this point clear to you all? Yes or no? And then another factor which affects enzyme activity is the presence of certain chemicals which are known as inhibitors. As the name says, it will inhibit the process. It will shut off the enzymatic activity. Now we have basically different types of inhibitors, but the one which is mentioned here is only competitive inhibitor. Now what does competition mean? So two people fighting for the same thing, right? That is what competition is. Now say this is the enzyme molecule, this is the substrate, and this is the inhibitor, competitive inhibitor. Now both competitive inhibitor and substrate, they are fighting for the same thing, that is active site. Let's take an example. Let's say this enzyme is succinic dehydrogenase. Now the succinate is the usual substrate molecule for succinate dehydrogenase. But now malonate comes in. Now in the presence of malonate, succinate cannot bind. It will be the inhibitor that binds in. It is competitive inhibitor. It is more competent. It is more, let's say, it is more efficient, and it is going to bind and not allow the substrate to bind. And if competitive inhibitor binds, the process will stop. So this stops the process. So substrate cannot bind and the enzyme action declines. For example, inhibition of succinate dehydrogenase by malonate. This is a competitive inhibitor. Why are the two able to compete? Why malonate is able to bind? Because they both resemble in their geometry. They resemble in their structure. Now, such competitive inhibitors are also used to control the bacterial pathogens. So, sulfa drugs, these are medicines used to control bacterial growth. These sulfa drugs are also competitive inhibitors. Please tell me that these points are clear to you all. Yes, guys, please tell me these points are clear to you all. Yes or no? Right? So, this was a question of 2024. The inhibition of succinate dehydrogenase by malonate, that is by competitive inhibition. Last not the least, we have classification. So enzymes have been classified into 6 categories. How do we remember? We remember it OTH lil. You have to remember it in order. OTH lil. See, oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases. This year also we've got a question from this. Every year we get one question at least from this topic. Now see, or we call them dehydrogenases. Just see the reaction. So they are going to catalyze oxidation reduction reactions. This was reduced, this was oxidized, now it will alternate it. Transferases are going to transfer the group. So there is this G group which will be transferred from S to S dash. Hydrolases helps in hydrolysis, that is breakdown in the presence of water, right? So hydrolysis, all your digestive enzymes are hydrolases. Now see, lyases, they also break down, but not in the presence of water, in the mechanism other than hydrolysis, so they leave a double bond. So you should know these equations also. Okay, then isomerases, they help in interconversion, that is isomerism, and ligases are going to help in linkaging of the two molecules or compounds. Please tell me these points are clear. Please tell me these points are clear. And then coming on to cofactors. Now, see, there are some enzymes which are simple enzymes. Simple enzymes are only made up of proteins, while there are other enzymes which require a non-protein part along with the protein part. Please tell me. Is that clear? So non-protein part along with the protein part. Now these non-protein parts are known as cofactors. You remove the non-protein part or the cofactor, this protein part cannot catalyze. Protein part is known as is known as apoenzyme, and together they are going to form the whole enzyme, so call it holoenzyme. Is that clear? Now these cofactors can be of three types: organic, inorganic, organic but tightly bound, then it is a prosthetic group; organic but loosely bound, it is a coenzyme; and inorganic will be metal ions. Are these points clear? I hope the flowchart makes it easy. I hope the flowchart is making it easy to remember. Yes, now you should know the examples here. Very important. See, already told you prosthetic, organic, tightly bound. For example, for peroxidase and catalase, we have heme. Heme is the prosthetic group. So if you remove the heme part from these two, they will not be able to function. Then coenzymes, again organic but only transient, means temporarily bound, loosely bound. And for example, coenzyme nicotinamide adenine dinucleotide, that is NAD, and NADP contain vitamin niacin. Why do you need to eat vitamins? They act as coenzymes. Some of them are. And then we have metal ions, for example, zinc. Zinc is going to catalyze the activity of carboxypeptidase, which is a protein digesting enzyme. See, it is lost when the cofactor is removed, so it is crucial in the catalytic activity. Please tell me that this point is clear to you all. And see, this was a question in 2024. Please tell me guys, the topic is done. Now we are going to move on to cockroach. Quickly give me a thumbs up. Yes, please. Quickly give me a thumbs up. Quick, quick, quick. Let's have a quick water break. फ्यूचर डॉक्टर नहीं सोते हैं, बिल्कुल सही बात है. All okay? All good? Are we able to revise everything in detail? How many of you feel that this is all in detail? Write down detail in the chat. This is all detailed? Yes, please. All in detail? Perfect. Mock test question papers, guys. You will find all the mock test papers if you take this Prime membership. Almost free, ₹1. You will get all the 700 lectures till your Re-NEET exam. You will have that validity period. Till your Re-NEET exam, you will be able to watch everything. So we understand that system has mocked us, but we are with you. Don't worry. You will get all the mock test series, absolutely. You will get all the tests. You will get many question papers for practice. Just dinner break or what? Dinner? No. No dinner break, please. How can we take dinner break here? I can just do one thing. See, let's revise cockroach and frog quickly. We'll be able to complete it quickly. Don't worry. Because I've already made short notes, and beyond this, you don't need to study anything for cockroach and frog. Trust me. Okay. And then talking about animal tissues. Otherwise also, I already planned that when I'm going to discuss human physiology, I am going to discuss animal tissue there. Muscular tissue in movement and locomotion. Neural tissue in your nervous system. I'm going to talk about epithelial tissue in the entire human physiology. Right? And connective tissue also. So don't worry. We have to revise cockroach and frog. Quickly. Now see, what is the classification? Animalia, Arthropoda, Insecta, Blattaria, Blattidae, Periplaneta and Americana. Is it clear? This is the classification. Size of cockroach is 34 to 54 long in adults. Colors, you get multiple colors. We in India see only the boring color, that is brown. Otherwise it is bright yellow, red, green and brown and black. Habits, they are nocturnal. They come out during the evening time or the night time. They're omnivorous. They eat both veg and non-veg, and they're found in the damp places near the drains, right? Now see here. Then what is the characteristic feature? Now being an arthropoda, it has an exoskeleton which is hard and made up of chitin. Now that exoskeleton is in the form of plates, and we call those plates as sclerites. We call these plates as sclerites. Exoskeleton plates are known as sclerites. Is that clear? Now see, each segment, say, let's say, okay, I'll use this as a cockroach. Now this is my white cockroach. Now say this is a cockroach. Now this is one segment of cockroach. So it will have a plate on the dorsal side, on the ventral side, and the two on the lateral sides, right? So each segment is basically surrounded by 4 sclerites. Now this is on the dorsal side, known as tergite. The ventral side is known as sternite, and the sclerites on the lateral sides are known as pleurites. So there are 4, 1 tergite, 1 sternite, and 2 pleurites. Please tell me that these points are clear. Please tell me these points are clear. Right? Now, arthrodal membrane. How are these plates attached to the body? So in between the plate and the body is present an arthrodal membrane, or we also call it articular membrane because it helps in articulation, means joining. Now this membrane is flexible. This membrane is flexible and it keeps the body flexible for breathing. Why do we need a flexible membrane here? See, this body, if it will be surrounded by tight, hard plates, how will it move? Inhalation, for inhalation and exhalation, it can't. So we need a flexible membrane to join the chitinous plate, that is sclerite, and yet keep the body flexible. Is that clear? Now these are NCERT lines. Now moving on to the body segments. Body is divided into 3 segments, that is head, thorax, and abdomen. So do you see this NCERT diagram? You see that? So till here we have head. What is present on head? We have 2 compound eyes. We have 2 antennae, which are these filiform structures, right? And then it has mouth, which has 5 parts, and it has ocelli. Ocelli are 2. Now ocelli are basically the simple eyes. They don't form image, but they check the intensity of light. Then on thorax, now do you see? This whole portion represents the thorax, right? So thorax has wings 2 pairs and legs 3 pairs. Is that clear? So this is pronotum, this is mesothorax, this is metathorax. Now on each segment of thorax, there are 3 segments, pro, meta, meso and meta. On each part of thorax, we have 1 pair of legs. Is that clear? So accordingly, we'll have prothoracic leg, we'll have mesothoracic leg, and we'll have metathoracic leg. Anyways, and then this boat-shaped structure, this is the abdomen. The 2 appendages which you will find on abdomen, anal cerci 2 and anal styles again 2. I told you arthropoda is arthropoda because of jointed appendages. Have we written the names? Legs, antennae, and anal cerci. These are all jointed. Jointed, jointed, and jointed. These are also jointed. These are the only appendages which are not jointed. These are unjointed anal styles, and they're only found in males. We'll talk more about them one by one. Please tell me that these points are clear. Please tell me these points are clear to all. Yes, please. All right. Now moving on to the head. See the structure of head. Do you see these jointed structures? 1, 2, 3, and so on. These are antennae. These are antennae, right? How many? 2. At the base of the antennae, do you see this structure? This is the ocelli, which represents a simple eye. And behind that, they have large compound eyes. Then there is this mouth. Now we'll talk about it one by one. Coming on to shape. First of all, the head is triangular in shape. And head is made by the fusion of 6 segments. Is that clear? Now position, head is at the right angle to the body. So let's say if this is the triangular, this is the triangular head and this is the body. So it is at right angle. So such is, and it is downwardly directed. It is at the right angle and mouth is downwardly directed. This downwardly directed mouth is downwardly directed. This is head, this is the thorax. Downwardly directed mouth is known as hypognethus. Hypo means below, gnetha means basically jaws, but we'll use it as a mouth here. So downwardly directed mouth. Is that clear? A right angle to the body, downwardly directed hypognathous. Now what are the structures that you see here? We see compound eyes which are large, 2 in number of course, kidney shaped eyes, and each eye will have how many? Each eye, so see, this is the eye, and each eye will have such hexagonal units, and these hexagonal units are known as ommatidia. These are known as ommatidia. Is that clear? Then it has sensory receptors called antennae, and these antennae are meant for smelling. So they keep them and these antennae in the air, and they're trying to find out the smell of food. So to find the food, they use these antennae. And again, these antennae are also jointed, just told you. Then about the mouth parts. What type of mouth are? They bite and they chew. So biting and chewing type of mouth. Now each mouth will have 5 different parts. So it has upper lip known as labrum. It has lower lip which is known as labium. One upper, one lower. Then it has mandibles in a pair, maxillae in a pair. Now mandibles have grinding and incising regions. Do you see? You should be able to label them. This is the upper lip. This is the lower lip. This is the tongue, that is a hypopharynx present within the mouth cavity, and these are the maxillae. So which ones are paired and unpaired? So these three are the unpaired ones, right? Unpaired. One labrum, one hypopharynx, one labium, while the mandibles and maxillae, these are the paired ones. Do we see that here? Will we be able to identify them? Please see, will we be able to identify them? Yes or no? Cutting and chewing type is also fine. Biting and chewing or cutting and chewing, both will be fine. Now moving on to the next, that is thorax. See here, talking about thorax. So thorax we know consists of 3 parts: prothorax, mesothorax and metathorax. Now head and thorax are basically connected through a neck. Now neck is an extension of prothorax only. Neck is the extension of prothorax. It is not a separate structure. Now what are the structures that you see on the thorax? I told you 3 pairs of legs. All the legs are similar. Do you see? On this. All the legs are similar. Just the size will be different. Now each leg will have 5 parts: coxa, trochanter, femur, tibia and tarsus, in this order. What are the legs meant for? Since it is a cursorial, it is a very fast running. So it is meant for fast running. It walks, and on the legs are present some taste receptors also. Okay. Now on the thorax are present wings. See, like we have seen that on every thorax there is a pair of leg present. In case of wings, guys, wings are only present on the meso and meta. There are no wings on prothorax. There are no wings. So that is why we have mesothoracic wing and we have metathoracic wing, because it arises from metathorax. So mesothoracic are the 4 wings, also known as tegmina, or we call them elytra, and then we have hind wings. Now you have to remember, 4 wings are opaque, dark, leathery, cover the hind wings, so they are meant for protection, not for flight. These are protective, while hind wings are meant for flight. That is very, very important to remember. Is that clear? Then talking about the abdomen. Abdomen is boat shaped. It is boat shaped, and it is made up of 10 segments in both males and females. Males and females are separate, and there is sexual dimorphism. Now see, the 7th sternum is boat shaped. Overall the shape is also boat shaped, but 7th sternum is clearly boat shaped in females. 7th, 8th and 9th sterna, they will form the genital pouch. These are all points of sexual dimorphism. Is that clear? What is the genital pouch? Where female gonopore and spermathecal pores and collateral glands will open. Now in males, genital pouch is at the hind end, and it is formed dorsally by the 9th and 10th terga and ventrally by the 9th sternum. So this is sexual dimorphism. Another feature, see, On the abdomen, we have anal cerci. Now, these are present in both the sexes on the 10th segment. And what are they meant for? Anal cerci have audio receptors, means meant for hearing. So, they hear sounds. So, sound sensitive. A pair of jointed filamentous structures on 10th segment in both the sexes and also on abdomen. But only in males are present on the 9th sternum, anal styles. They are only in males and helps in distinguishing the sexes. They are unsegmented. What are the function of anal styles? They help in holding the female. Please tell me that these points are clear. Now talking about the digestive system. Very clearly, do you see the diagram here? So it starts with the mouth, goes to the pharynx. This is the pharynx shown here. Then the esophagus, which is short, and then opens into this dilated region which is called crop. So crop and gizzard, not only in birds, it is also found in cockroach. Clear? Now, esophagus, crop always stores the food. Gizzard, also known as proventriculus, it has 6 cuticular or chitinous teeth and they are meant for grinding food, and that is why we call gizzard as a true stomach. This is where the food is grinded properly. Is that clear? Now, this structure, this is the gizzard. Now, beyond gizzard, we have this structure which is called midgut, which is called midgut. So, midgut is also known as mesentron. It is also known as mesentron. Is that clear? Now, at the junction of foregut and midgut, do you see? At the junction of foregut and midgut, we have these structures. These are known as hepatic caecae or gastric caecae. Caecae because they are blind from one end, they are closed from one end. And what do? They secrete the digestive juice. Already told you, at the junction of foregut and midgut. Then we have hindgut. Beyond this, we have hindgut made up of 3 parts: ileum, colon and rectum. Are these points clear? Okay. Now hindgut is broader than the midgut and includes these parts, and they open in outside. Please tell me that these points are clear to you all. Please tell me these points are clear to you all. Quick. Please tell me these points are clear. Moving on. Okay. Now, talking about the blood vascular system. So, being an arthropoda, see all the arthropods, they have open circulatory system. So does this. And open circulatory system means no capillaries. So, blood will fill in the sinuses. It will fill all the sinuses, and that is why the body cavity will here be known as the hemocoel. Body cavity is known as the hemocoel because it is filled with the blood. Blood is known as hemolymph. It is white in color. It is whitish because of no respiratory pigment. It is colorless. Now, what is the blood made up of? The cells called hemocytes and plasma. These hemocytes are more like WBCs. They are phagocytic in nature. Is that clear? Then heart in non-chordates, it is dorsal. It is muscular tube like on the mid-dorsal line, and it has 13 funnel-shaped chambers. Do you see these? These are the funnel-shaped structures, chambers of heart. And how many chambers are present? 13 chambers. It is a tubular heart. On which side? On the dorsal side. Is that clear? Now, it looks like this actually. It looks like this. This is a funnel-shaped heart. So, on the sides, we have the openings through which blood is going to enter, and these openings are known as ostia on both the sides. So, blood from the open spaces, so this whole space is filled with the white colored blood. So, blood is going to enter from here into this, through the ostia into the heart. And what is the direction of blood? Blood goes from posterior to anterior direction. From sinuses into the heart and interior, so from posterior to anterior, and it goes into the interior aorta. So this is how the blood flows from sinuses, heart and sinuses again. Is it clear? From heart to aorta. So here comes aorta and then to the sinuses again. Please tell me that these points are clear to you all. Is that clear? Now, there is an important structure. Do you see that there are these funnel shaped, these triangular muscles present at the junction of two chambers? So we have 13 chambers and we have 12 pairs of alary muscles. These alary muscles will move like this. They are going to help in the contraction and relaxation of the heart, alary muscles. Is that clear? Now coming on to the respiratory system, the respiratory structures used by cockroach are known as trachea. So these are the trachea. Do you see? These are the openings present on the side of the body called spiracles. There are 10 pairs, 10 on each side. 2 pairs are thoracic and 8 pairs are abdominal. Is that clear? Is that clear? Now, these are the holes through which air is going to enter, and this whole tube is known as trachea. This is called trachea. Now, like our trachea has cartilaginous rings, these trachea have cuticular thickenings. Again, for the same purpose, they prevent them from collapsing. Prevent the collapsing of, of trachea. Are these points clear? So blood is going to directly diffuse. So from this atmosphere through spiracles into the trachea, trachea will break into these tracheoles, and tracheoles are surrounded by the tracheolar cell, and so air directly from the trachea goes into the cell. This is called direct respiration. Tissues direct in contact with the air. Is that clear? Now spiracles are not always open, few are, but spiracles are also regulated by sphincters, so that they are not always open. If spiracles will always be open, water will be lost, dehydration will take place. I hope these points are clear to you all. So these are the respiratory structures. Moving on to the excretory, they have characteristic structures called Malpighian tubules. Now Malpighian tubules, we have seen the structure at the junction of midgut and hindgut. At the junction of foregut and midgut is gastric caecae or hepatic caecae, while midgut and hindgut will have hundreds or 150 in number. These are Malpighian tubules. Do you see these are yellowish colored bodies? Yellow, 100 to 150. So what do they do? So as we know, all around them is present the hemolymph. So from hemolymph, they will absorb the nitrogenous waste and convert that nitrogenous waste into uric acid. So it is uricotelic. So other than Malpighian tubules, we have other excretory structures like fat body, nephrocytes, uricose gland. Now this is a PYQ, guys, already. Which of the following are the excretory structures in cockroach? So you should know these names. Are these points clear? Now, what happens? Nitrogenous waste from the hemolymph will be collected, converted into uric acid, then poured into the alimentary canal. So alimentary canal not only helps in digestion and release of the waste food material, alimentary canal will also remove the uric acid. Please tell me these points are clear. Please tell me these points are clear to all. Yes, please. Anyone with any doubt? And now we talk about nervous system. We talk about nervous system here. Now see, nervous system in non-chordates. First of all, I told you, nerve cord will be double, ventral and hollow or solid? Solid. Right? Now see, it consists of fusion of double. Do you see there are two pink lines I have drawn? Right? So it is double ventral nerve cord. Is that clear? Now there are so many ganglia present. So you see around 9 ganglia present. 3 ganglia in the thorax region, 6 in the abdomen. Is that clear? Now this nerve cord, these two nerve cords start from the brain. They start from the brain. Now brain is represented by these two supraesophageal ganglia. It is represented by one supraesophageal ganglia. So let's say if this is the esophagus and on the top of esophagus is present a ganglia. Ganglia is a group of cell bodies basically. Okay. Now supraesophageal ganglia represents the brain. Now this supraesophageal ganglia or the brain is from where double ventral nerve cord arises. And do you see there are 3 thoracic ganglia and then 6 abdominal ganglia. Is that clear? Is that clear? Now, other than brain and this double ventral nerve cord, we have sense organs, which we have already discussed, antennae, compound eyes, and so on. Is that clear to all? Please tell me these points are super clear. Now, the most important system, which is the reproductive system, guys. Now, see, talking about the male reproductive system, this is the structure. First, let's discuss the primary structure, that is testes. Now, it consists of 2 testes, 1 and 2. Do you see? Now, this starts from 4th, 5th, 6th. So, testes are present between 4th, 5th and 6th segment. They are long, they are tubular and they are follicular. They are long, they are tubular and they are follicular. Is that clear? Now, the function of testes is of course to produce sperms. Now, from these testes arise these structures. Do you see here? What are these structures? These structures are vas deferens. So, vas deferens are going to carry the sperms from testes to the seminal vesicle. These are the seminal vesicles. Is that clear? So, from the testes, sperms are going to enter, see, testes to vas deferens. Vas deferens carries it to the seminal vesicle. Now, what is the function of seminal vesicle? Seminal vesicles are going to bind the sperms to form a ball-like structure. Is that clear? So, these are going to form the sperm ball. See, they're going to bundle them together to form ball-like structure, or we call them spermatophores. Sperm bundles will be formed by seminal vesicles. Is that clear? Now, do you see that there are some long and small tubules? These together form the mushroom gland. Now, these are the long and the small tubules. Is that clear? Now, the long tubules, they are going to secrete a membrane. See, around what happens here? So, here in the seminal vesicles, guys, sperm balls are formed. Mushroom gland is going to secrete and form them further sperm balls, and from the ejaculatory duct. Where is the ejaculatory duct? Do you see here? This is the ejaculatory duct. So, the sperm balls will be put out of the body through the ejaculatory duct. Is that clear? Now, along the ejaculatory duct, do you see there is opening? Now, here we have another gland, which is the phallic gland, also the Congo plate gland. And this is the duct of the Congo plate gland. So, while the sperm balls will be brought out here, the Congo plate gland will also produce a secretion. Is that clear? So, this is how. So, ejaculatory duct and then Congo plate gland or the phallic gland, that will also pour a secretion and near the male genital pore, and now the sperms will not be released outside. See here, it has these structures, left phelomere, right phelomere and ventral phelomere. Now, these phelomeres, these are chitinous structures. They are asymmetric, no fixed shape, asymmetric, and they act as external genitalia. So, these phelomeres are going to open the female genital pore. They are going to help in opening the female genital pore so that the sperm balls can be released in the female body. Please tell me that these points are clear. Now, see here. This is the female body. Do you see this structure? Do you see this parachute-like structure? This is the spermatheca. There are 2 spermatheca, one on this side, one on the other side. This is the place where sperms are stored. Sperms are released or sperms are stored. So, the male cockroach is going to open this female genital pore and release the sperms in this structure. Is that clear? Okay. Now, coming on to the female structure. So, females have these ovaries. Females have 2 ovaries, 1 and 2. Each ovary has around 8 ovarioles. So these long structures, this is one ovariole, this is another ovariole, and so on. So each ovariole will produce one egg. So how many total eggs will be produced? 8. 8 from this side, 8 from this side. Is that clear? Now from ovariole, see here, lies this oviduct. So eggs or ova from the ovarioles enter into these oviducts, and together they come to lie in this common oviduct, also known as vagina. And in front of the vagina is present, in front and behind the vagina are present these spermathecae. Please tell me these points are clear. Please tell me these points are clear. Yes? Right? So these spermathecae are present. Clear? Now what will happen? Now the eggs will be released here. The eggs will be released here in the genital chamber. And when the eggs are released here, the spermatheca will contract and they will pour the sperms. So basically, where does fertilization take place? Fertilization is internal. Where does fertilization take place? In the genital pouch, site for fertilization. So basically, it is the internal fertilization that takes place. Internal fertilization. Please tell me these points are clear. Please tell me these points are clear. Now, do you see that there are these branched structures present here? These highly branched structures, these are the collateral glands. Now, once the eggs have been fertilized, these collateral glands are going to pour a secretion and form a reddish brown capsule, which is known as ootheca. It is known as ootheca. Is that clear? So they are encapsulated now. The fertilized eggs will be protected within a capsule, and that capsule is known as ootheca. Who secretes that? Collateral glands. They are branched. Is that clear? So they contain fertilized eggs, and then females will produce around 9 to 10 ootheca in their entire life, and each ootheca will contain 14 to 16 fertilized eggs. Please tell me that these points are clear. And then development is indirect. It will have a larval stage which is known as nymphal stage, and that metamorphosis is known as sporometabolous. Nymphs look like adults, but they will not have wings. It has to undergo 13 moltings to form the adult. Now this line, next to last nymphal stage, what does it mean? Second last, not 13th, but the 12th one. So next to last nymphal stage will have wing pads, but wing pads are different from wings. Only the adults will have wings, not the larva. Please tell me that these points are clear to you all. Yes, please. Yes, guys, please let me know that these points are clear to you all. Yes or no? Can we start with the last thing of the day, of tonight, let's say? Yes, guys, quickly let me know. Can we start with the frog? Give me a thumbs up. Are we all awake? Are we all energetic? Are we all ready to learn the last topic of the day, that is frog, Rana tigrina? There were 3 questions. Yes, guys, quickly, the last topic, quick. Yes? All right, absolutely. Now, frog, which is the Rana tigrina. Rana is the genus, this is a species. So we know the classification: Animalia, Chordata, Vertebrata, Gnethostoma, Tetrapoda, and Amphibia. We have read the animal kingdom today itself. Now see here. Okay, just a second. Yeah, now see here. What is the skin like? It has a smooth, moist, glandular skin. We have read in amphibians. Mucus is present. Now, these are the adaptations for cutaneous respiration. These are the adaptations for cutaneous respiration. Yes or no? Now, the upper side of the skin, dorsal side, it has olive green with dark spots. In the ventral side, the lower side is pale yellow. Now, water is absorbed through skin. It never drinks water. Interesting. The body's division made up of two body divisions, head and trunk. Remember, trunk is a combination of thorax plus abdomen. But when they are not distinguishable, we call it trunk. Neck is absent, tail is absent. That is why it belongs to the category of Aneura. Frog doesn't have, but tadpole has a tail. Now, talking about head features. Now, what does head consist of? It has external nares, that is nostrils. Eyes are bulging, they are large, and it has nictitating membrane, which is the third eyelid. Nictitating membrane in our case is vestigials. You know, the inner pink area, that is nictitating membrane. It is vestigial, but it has a functional nictitating membrane. Tympanum represents the external ear because there is no pinna. Limbs are present. See, limbs are present, 4 limbs, 4 digits, no thumb. Hind limbs will have 5 digits, larger and more muscular for leaping. So it is with the help of the hind limbs that it actually jumps. Now, the hind limbs will have webbed feet. Now, digits of hind limbs are joined with the webs. These are the webs. And why do they have webs? For swimming. Now, we see clear-cut sexual dimorphism in case of frogs also. There are 2 features of sexual dimorphism. Males will have vocal sacs and they will have copulatory pads, which will be absent in case of females. Vocal sacs only during the breeding season, which will develop for the croaking sound, mating call, and copulatory pads only on the first digit of the 4 limbs, again during the mating season. Is that clear? Now, this copulatory pad will have suction pressure to hold the female during copulation. So that is why only males will have. Is that clear? Now, talking about the digestive system made up of alimentary canal and digestive glands. Alimentary canal is complete here. See, starting with, let's make it little small. Okay, yeah. Now, see here. The mouth, alimentary canal starts with the mouth, buccal cavity, pharynx, esophagus, stomach, intestine, rectum, and cloaca. Remember, there was a question, cloaca, and we have read the line also in amphibians. Alimentary canal, reproductive, and urinary or excretory. They will all, these openings will open together in cloaca. Clear? Then talking about the intestine. Intestine is short. Intestine is always short in the carnivores. This is a modification. Carnivorous habit, it has a short intestine. Digestive glands, guys, now talking about the digestive glands, consist of liver and pancreas. Liver, just like in our case, it secretes bile, which will help in the emulsification of fats. Pancreas will secrete the pancreatic juice. Now, talking about digestion, it has a tongue also, bilobed tongue. And that tongue will be sticky. The tongue is sticky, which will help in capturing the prey. So it takes its tongue out, the insect sticks to it, it brings it back, and it's very quick. Now, stomach will secrete HCl and the gastric juices to form the chyme. Duodenum, that is the intestine, receives the bile and the pancreatic juice. Bile will help in emulsification. Pancreatic juice will help in the digestion of carbohydrates and proteins. Please tell me these points are clear. I have told you, Jyoti, the tissue part will be taken care of along the human physiology. So everywhere I am going to discuss the tissues there. Is it clear? All right. Moving about absorption, it occurs in the intestine, and for increasing the surface area, we have villi and microvilli. Egestion occurs through rectum, and rectum will open into not outside but cloaca, and then cloaca will open outside. Cloaca is a common chamber. Is that clear? Now, moving on to the, see, digestive system has been shown here. Do you see? This is the esophagus. This brown structure shows the liver, bilobed. Then this is the elongated stomach. This is the intestine, which represents the duodenum, right? Then it has a rectum, and then rectum will open into the cloaca. Is that clear? So in this diagram, they have shown the digestive system also. Clear? All right. Moving on to next. This is the respiratory system. Respiration I've already discussed. Now, in the larval stage, larva will respire through gills. That respiration is known as branchial respiration. While the adult will have cutaneous, pulmonary, and buccopharyngeal respiration. So, through skin underwater, as well as during hibernation and estivation, it is going to respire through skin. But otherwise on land, it will use lungs and it will use a mouth, that is buccal cavity. Now there are pair of sac-like pig structures known as lungs. Is that clear? Talking about the circulatory system. Now see, very important. Circulatory system is of closed type. In cockroach we have open type. Here we have closed type. Heart is myogenic and ventral. What is myogenic? So there is a SAN, there is a sino-auricular node which is going to produce the impulse, and it is on the ventral side. This is a chordate feature. Now we have read amphibians have 3-chambered heart, 2 atria, 1 ventricles. There is a protective covering known as pericardium. Is that clear? Now see, the heart has right auricle, right auricle, left auricle, and the common ventricle. Is that clear? I'll talk about these things in a while. Now blood consists of the liquid part known as plasma, and it has cells, RBCs, WBCs and platelets. RBCs are nucleated and it has hemoglobin. Now our RBCs don't have nucleus, but their RBCs have. Is this clear? So this is the cellular part. Now the double circulation, because the blood is going to enter the heart twice. First, in this side it is deoxygenated blood. This side we have oxygenated blood. Is that clear? Now, see, directly the blood is not going to enter into the right auricle. Let's see the structure here. Let's see the structure here of the heart. So this is the dorsal view. Now, do you see, this is the superior vena cava. This is another superior vena cava, we call them precava, and this is inferior vena cava, also known as the post cava. Now 3 vena cavas, they open in this common chamber which is known as sinus venosus. Is that clear? This is on the dorsal side. This is the dorsal side. So from the dorsal side, the sinus venosus collects the deoxygenated blood, and now this sinus venosus is going to open into the right auricle. Is that clear? So directly the vena cava doesn't open into the right auricle, it opens through sinus venosus. Are these points clear? Here we have the left ventricle. From both the sides, the blood is going to enter into the common ventricle. This is right auricle, left auricle and the common ventricle. Mixed blood. Mixing will take place here. Now, do you see this red structure? This structure is called truncus arteriosus. This structure is known as truncus arteriosus, which is going to take the blood outside. Is that clear? Which is going to take the blood outside. Now talking about the excretory system. Wait guys. Yeah. Now see, the excretory system consists of kidneys, ureters, urinary bladder and cloaca. Kidneys are paired, dark red, bean shaped, and the functional unit of kidneys will of course be nephron. Now in case of males, now this was the line. This was the NCERT line, and this was exact line, isn't it, in NEET 2026. This was the exact line in NEET 2026. Did you see? Ureters and oviducts open separately in females, while in males, ureters are the common ducts which will carry both urine and sperms. So we call ureters as urinogenital ducts. They gave this line in NEET 2026. This can be in Re-NEET, maybe. Yes, can we predict that? Now excretion, the nitrogenous waste is urea. So it is ureotelic, while cockroach was uricotelic. Is that clear? Now talking about the nervous system. Nervous system is divided into central nervous system, peripheral nervous system and autonomic nervous system. Do you see? The central nervous system consists of brain and spinal cord. Peripheral nervous system has cranial nerves and spinal nerves. And how many? That is 10 pairs, right? And then we have autonomic nervous system. This was the question this year. There were 10 pairs of, they had written there were 12 pairs. Our correct answer is there are 10 pairs, not of cranial, spinal, but of cranial nerves. We have 12 pairs of cranial. They have 10 pairs. Is that clear? And then autonomic nervous system always deals with the involuntary structures. Now see here, the brain is covered by cranium, that is the brain box, and then there are some coverings known as meninges. We have 3 meninges, they have 2 meninges. They have dura mater and pia mater. We have an additional, that is the arachnoid. We'll talk about it tomorrow. Then forebrain has olfactory lobes 2, cerebral hemispheres 2, diencephalon 1, just like us. Midbrain consists of 2 optic lobes. How many optic lobes do we have? 4. Hindbrain, cerebellum and medulla oblongata. There was this statement that hindbrain has cerebellum, medulla oblongata and 2 optic lobes. That was wrong. This was in NEET 2026. You should know which part comes under which type. This was all in NEET 2026. Yes or no? Yes or no? These lines were there, right? Then moving on to sense organs. Let's talk about the sense organs. So for different sensations like touch, it has sensory papillae. Taste, it has taste buds. Smell, it has nasal epithelium. For vision, it has large eyes. And for hearing, it has tympanum and the internal ear. Is that clear? Is that clear to all? Now talking about the male frog. Male frog will have testes, which are yellowish ovoid bodies attached to the kidneys, and by a double fold of membrane or peritoneum known as mesorchium. So mesorchium connects the kidneys to the testes. Now there are 10 to 12 vasa efferentia which will enter the kidneys and open into the Bidder's canal. So from the Bidder's canal is going to carry the sperms into the urinogenital duct, which is the final exit for sperms. So from there it will be urine or the sperms, and it opens into the cloaca and then outside. Then female, it produces eggs in the ovaries. What is the egg capacity like? 2500 to 3000 eggs in a day at a time. So it is paired and near kidneys, oviducts open into the cloaca and then the outside. So do you see, both male and female, they are releasing their gametes outside. So what type of fertilization? External, that is in water. It is going to produce a tadpole, hence indirect development. Please tell me that these points are clear to you all. Please tell me. Yes guys, is that clear to all? And so guys, with this we complete today's agenda. We have completed animal kingdom, we have discussed biomolecules, we have discussed frog and cockroach. As far as animal tissue is concerned, we are going to discuss animal tissue along with human physiology. We will see at every place what type of tissue is present. Is that clear? So give me a thumbs up in the end that we have been able to achieve today's agenda. And guys, tomorrow 7 p.m. I'll be meeting you again with all the chapters of human physiology. And now don't forget to subscribe to the Prime, right, for attending all the tests, for taking AIMT, and also for watching all the lectures till your NEET exam. All clear? So we have revised class 11 part 1 zoology. Yes? Perfect. All right guys, thank you so much. Now don't lose heart. Keep this energy high till the NEET exam and make it even better than NEET exam. Is that clear? It is all about perspective. I'll see you tomorrow with the same energy and we'll be discussing human physiology. Thank you guys. Good night and keep revising.